Hello and welcome to Apologetics 101. I'm your host Brian, your resident Christian apologist. Today we're going to be doing another Are We Apes, or another video in the Are We Apes series, and we're going to be featuring Matt and Donnie from Standing for Truth Ministries, and they're going to be both on here, but originally they were supposed to be on here together. And Donnie made a mistake on when it was scheduled because we were going around with a few different dates. And so he had it scheduled down for the 5th when it was supposed to be the 1st. And so we end up doing, me and Matt went ahead and did our interview on the 1st. And then me and Donnie did our interview on the 5th. And then I decided to splice it together. So that's what I'm going to do. I'll probably add an explanatory comment in the middle just to kind of explain a little bit between the two different interviews. And so I interview Matt and then I interview Donnie, but it's all going to be part of one video. So without further ado, let's dive in.
Hello, my gentle and, of course, very modern apes.
Sure, I'd like to kiss you goodbye.
All right, but you're so ugly.
Human beings are not just related to apes, but actually aren't.
This is a primate, and so is this, and so is this, and so is this. Hey, that's me.
We are still hominids. We're still primates, mammals, tetrapod.
Well, hello and welcome to Apologetics 101. I have for the first time ever on this channel, Matt. He's known online. Is it okay to mention your last name? I saw your last name on Christian Today. I don't know if that's something you want known or not. I just want to make sure.
It's fine.
Okay.
Yeah.
Matt
Yeah, I'm
Nailor.
doxxed already, so it doesn't matter.
Right. But you are known online either as Mattman or as Raw Matt. And I've always wanted to ask you, how'd you get the name Raw Matt? I'm just curious. Is that a story I want to know about?
That's an interesting one. Yeah, in 2002, I became a raw food vegan, and I kind of came up with this concept of like, hey, since I only eat raw, it kind of like matches, kind of flows really good. So I just stuck with it because that was my diet for so long.
Oh, and everybody else stuck with it too, because even some critics call you Raw Matt.
Yeah,
But
yeah, they like it. That's how they found me.
right, right. Um, And well, we usually start with an introduction. I know this is your first time visit to this channel, and I know you're part of the Standing for Truth ministry, and you also run the Young Earth Creationist YouTube channel as well. And well, you do your own stuff too, if I'm not mistaken. Am I correct?
Yeah, I had the Young Earth Creation channel before I even met Donnie,
Oh,
but it wasn't really about Young Earth Creation. It was on dietary health and nutrition and weird enigmatic topics, kind of cool things, very different. And I believed in evolution at the time. And then right before COVID hit, I started to look at Young Earth Creation. I was like, wow, I like this. Let me do some more digging. And then I jumped into it pretty hard because of all the free time, especially during COVID. And I just changed the name of my channel to Young Earth Creation. I couldn't believe it wasn't taken. I was like, "Are you kidding me? I got Young Earth Creation? That's awesome!" And then I found Donnie. He was dressed like SpongeBob, you know that little character in the stream, and he's debating people all the time. I'm like, "Who is this guy?" Like this guy's great. I want to form a team of people that all want to fight against these atheists, these God-hating critics online that say it's not true. And I'm like, you know what? I'm going to be the first to do it. I'm just going to grab people. I don't care what their denomination is. I don't care what they believe. We're just going to finally unite everybody together. And so that was the goal, and that's exactly what happened. And so we formed a team standing for truth, and so we made his channel the main one, and mine became backup because, as you know, YouTube, you can't really trust it very much. They take channels down all the time. So mine became a backup for Standing for Truth. And you're right, I do make my own content. I have my own books and stuff, but all together, we make similar things. I'm just a video editor, you know?
Right. Donnie mentioned that you edit his videos and stuff because he talked about how good I am on editing my videos because I do everything. I wish I had a team behind me sometimes to work a lot of
It's
the
very
stuff.
hard.
Yes, yes, it is. But I do my own website. I do the my own, which is up to me pay another because I have to pay that every year, and so it's up for renewal now. So I
Oh,
usually pay
no.
that with my income tax return, and I'm waiting for that now to come in, and I'm gonna take a vacation toward the end of
Yeah.
March. So that's why I haven't produced a video yet this year. So I'm pushing for a lot of videos all at once. So
Oh,
so pray
wow.
for me. But
It's good. Finally, another video editor, somebody that knows how hard it is. You are right.
oh
It is
yeah,
so time consuming.
yes, yes it
So
is.
time
Yes it is very.
consuming.
Yeah, that you have the Young Earth Christians channel, and it's now acted as kind of like a backup channel to Donnie's in case he's ever get taken off. And and you fight against skeptics. Oh, there was a question I had on your statement there. On you said that you used to believe in evolution. Was you a theistic evolution? Yes.
No, not because I wasn't a Christian at all, really. So, I was just a non-believer. I grew up with a family that were not religious, but they weren't atheists. They didn't care. You know, they're busy with work and life and having fun. So I just grew up around the same atmosphere. So I didn't really look into it. And when I got out of school, all I wanted to do was compete and train and fight in martial arts. So I got right into MMA, and that's the last thing on your mind. You know, you're just competing and training and staying at the top of your conditioning all the time. You don't have spare time to be like, gee, I wonder what happens when I die. You know, it didn't really cross my mind. So later, it took the evidence to actually convince me. I'm not like one of those people that had like an emotional epiphany that like, oh my gosh, I got in a car wreck and I saw heaven. I didn't see anything. I just studied it and I was like, this makes total sense. This is great.
Awesome. Um, and I actually used to believe in evolution a long time ago myself, but that was before I was a Christian. I started reading the Bible, and I realized the Bible did not support the idea of evolution. I realized you could not force millions and billions of years into that scenario. And I became convinced that originally I became convinced by the biblical evidence. Later on down the road, I started having my doubts. One of older creationism is true, and that sort of thing. And I started studying the evidence concerning uh, uh, young Earth creationism, and I realized that the evidence was very, very strong, and uh, and became much more convinced that the biblical creation perspective is indeed true. And so that was my my take on that because I do believe the Bible, the straightforward rendering of the Bible, clearly indicates the world is young and the evolution is false. This is not the way in which the Bible describes how God created everything. Um, so with that said. Okay, just to kind of explain to everybody, I know we already just kind of started this off with me and Matt already knowing what the situation is. Everybody was probably expecting Donnie to join us on here, but Donnie actually mistakenly thought it was the fifth. he put it down on his calendar as the 5th. And so me and Donnie, I guess I'm gonna talk to him to make sure that this is what he wants. But I'm thinking Donnie will um, join us on the 5th of this coming Wednesday, and he'll be able to relay his part. And then I'm gonna try to see if I can combine it in editing. So just to kind of let you know, Donnie or Matt's going to be giving his presentation today, and then I'm gonna ask questions to Matt based on what he presents today. So unless you want to add anything, Matt, before we begin, and you can share your screen whenever you're ready.
Okay. No, no, I like that. Yeah, I'm kind of the same as you. When I went to church, nobody ever even talked about creation or evolution, so it wasn't a topic that like came, up. So it could it took on being online and kind of like surfing around and seeing these things where it caught my attention. And actually leads us directly into the topic of the presentation, really. And that is, evolution firmly teaches that there was no Adam and Eve, and that there were no original two people, and that mitochondrial Eve, or the most recent common ancestor, doesn't refer to Adam and Eve. It just refers to pretty much anybody, the most recent common ancestor, definitely not the common ancestor to all people. So that's kind of what got me going, okay, well, what does it really mean then? Like, what happens when you type into YouTube, how old are humans? Well, it specifically tells you that they're old. They're about 300,000 years old. So that clearly is incompatible with what scripture tells us if we're going to go by the genealogies that take us back in the Masoretic text about 6,000 years, or the Greek Septuagint about 7,650. So that's a very short period of time compared to 300,000. And why would they actually say that if people are really not that old? Like, where are they getting this from? That was my whole question, because when you know that they're young, are they hiding the evidence? And that's what our talk today is going to be on. Do you think that'll be a fun one?
Oh yeah.
Well, for this one, we're just going to take a real quick dive into mitochondria. The mitochondria is something that is passed on maternally to parent to offspring, and so on. And every generation, we can count these mutations up. They're not like the next generation gets one and the next generation gets two. It's a little bit more convoluted with mitochondria. They don't pass on a single mutation every single generation. It's only about 16,569 base pairs long, so it's quite small. So it passes on a little bit less. So it's a little bit harder to count. But with evolution, what they do is they go, okay, well, we should be able to take this back in time to see what a most recent common ancestor is. It should go back a very long time, right? We're talking hundreds of thousands of years, right? That's what they said when you type it in online, right? I mean, 300,000 years is quite a long time. But we're saying, no, if we were to track these mutations back, we should find mitochondrial Eve. It should land on the biblical timeline. That would be the creationist prediction. If it falls outside of that, then it's clearly incompatible with scripture. So then they finally scanned the mitochondria, and they found that there were only 24 fixed substitutions. Now, what that means is that when a mutation comes up and it arises in the system, how long does it take before it reaches the pinnacle of fixation where everybody in that population now has it? For example, let's say that you lived in a population of 24 people, and somebody gets a mutation and a child is born with blue eyes. How long until that population all have blue eyes? Well, if it's a small population, it's not going to take very long. So evolutionists were shocked to find out how few total mixed or fixed substitutions there actually were. And they're really easy to see. So it's not like they were just guessing like, oh, well, maybe this is a substitution, maybe it's not. So they said, the populations must have just been very small in the past. They could have never really grown fast and rapidly, kind of like scripture says, a couple generations later, we were at the Tower of Babel and we started growing exponentially from then. So I guess it's really, how do you view the past? Are you looking at this mitochondria and seeing through the eyes and the lens of evolution? Because when you do that, you're setting your presupposition of belief on how you're going to read that data. How are you going to interpret what you're looking at? If your perception is only like yours and I's was based on evolution, then we're only going to see evidence that points to evolution because we can't think outside the box. It's really kind of a messed up paradigm that we're put into. So when they did that, they said, okay, well, we need to calibrate this to when man split from chimpanzees about 7 million years ago. So all they did is calibrate it to the fossil record. That's it. So they go, well, this is true. All these rock layers are absolutely true. So when we do that and we take that math and we run it back, the fossil record is now used to calibrate when mitochondrial Eve lived. So based on those 24 fixed substitutions, you get a mitochondrial Eve, the mother of all living, about 300,000 years ago. That's correct. But that, again, is assuming evolution. So what they expected to find when they eventually scanned the genome of living people, they expected to find one substitution every 300 to 600 generations from that split. That's what they determined based on the evidence of evolution being true. Well, the first study finally came out, in about 1995. And it was a D loop going back from 11 generations deep. And they found one substitution every 25 generations. You know how far away 25 is from 300 to 600? That's exactly the opposite of everything they expected to find. This actually puts the most recent common ancestor of all the European people, because that's what that study was actually done on, about 5,000 years ago. So all Europeans they would descend from a biblical line of one of Noah's sons, of course, and it fits right in the biblical date. Evolution would say that, wow, over 7 million years, that's a whole lot of mutations building up, isn't it? And they would be absolutely right. But that's not what we see. We find very few mutations, so few that it's actually perplexing. But then along comes Thomas Parsons. He's a forensic scientist that works for the FBI, and he has access to the blood bank. It's the largest in the world. So he decided to form a team and go through all of the data and run the first ever gigantic pedigree study to determine what is the most recent common ancestor and what is the actual mutation rate of human beings. So he took this and he looked at all the different ethnic people. There were 90 African Americans, there were 115 Afro-Caribbean, 114 Sierra Leone Africans, 90 Hispanic, 100 British Caucasian, 233 Europeans. So it was a mix, a random grab at exactly what you want. You do not want to just get mitochondria from a single line of people. And when he finally came out with his results, it was 10 substitutions every 327 generations, which is one substitution every 32 generations. Remember again, what evolution expected to find was the exact opposite, one out of every 300 to 600. And here we are finding rapid fixed mutation rates rapid substitution rate, really, really fast. So he published it, and the final conclusion of that paper in 1997 was a mitochondrial Eve that lived 6,500 years ago in the published peer-reviewed journals and literature. So when we go directly to the paper itself, it says, thus, our observation of the substitution rate is roughly 20-fold higher than would be expected from the phylogenetic analysis. Now remember, phylogeny is the assumption of evolution. This is the phylogenetic tree that they're assuming with the fossil record. So it became the exact opposite. It was 20 times faster, with the empirical age resulting in a mitochondrial Eve living 6,500 years ago, the biblical timeline. And then how do they get this rate? Well, you can see right here how it's all broken down if you want the technical breakdown of how to do it and how they were able to determine 6,500 years based on the maximum divergence differences between people in the world. And then we move on. Because the substitution rate equals the mutation rate, there is no way they can get around this now. It's obviously not exact, but it is very close. And this is used by the FBI today. It is in their forensic DNA application. So like, let's say that O.J. Simpson was tried for the murder, but what happened? What if his son had done it? Well, his son has the same DNA and blood type. So unless they had his son's DNA, it might have had an extra mutation in it that would have identified his son as doing it and not him. And that's why he got off, because they couldn't actually identify that it truly was his blood. And that's why he probably tried to send his son out of town, like right before this all happened. And the second they found the blood and everything, the first thing he did was buy his son a plane ticket out of country and do all his other things. Anyway, I'm pretty sure we know what's going on, but the DNA is what can convict people, and knowing this mutation rate is an actual very important thing that's used in forensics. But it gets more interesting because more people came along and started to look at this data, and we have an atheist by the name of Ann Gibbons, and she writes scientific papers from different studies, and she published in Science called Calibrating the Mitochondrial Clock. She wrote an article about these different studies. And she said, by the way, she uses the term evolutionist. So don't get mad at me. I know atheists hate when you call them evolutionist. I don't know why. I don't get offended when they call me creationist. But nonetheless, she's using the term evolutionist here, okay? She's referring to her own people as evolutionist. So he said, I could also complicate the lives of it could also complicate the lives of evolutionary scientists who use the mtDNA mutation rate as a clock to date such key events as when human ancestors spread around the globe. Evolutionists have assumed that this clock is constant and ticks off a mutation every 6,000 to 12,000 years. Think about that. In reality, they tick off a new mutation every 5 to 6 generations. They couldn't be more wrong. So regardless of the cause, evolutionists are most concerned about the effects of the faster mutation rate. So when you hear people say, oh, this doesn't bother us, this doesn't refute evolution, it absolutely does. And not only does it refute evolution, it confirms the biblical creation timeline and predictions, which is what we want to do. We don't care about mocking evolution or refuting evolution. We need to replace it with something better. And that's exactly what the biblical creation model does. So we jump into that article, the one that she wrote, and she said, using our new clock, she could be a mere 6,000 years old. I just love that quote. I like that. So I threw that in there. So when we go back to the actual study, what did she actually say about it? No one thinks that's the case. What do you mean? No one. Of course there are. Matter of fact, when this article was written, America was 50% young Earth creation. So clearly a lot of people thought that was the case. But of course, none of the people that write these articles in journals and scientific papers would ever believe that the mother of all living people lived 6,000 years ago. So his arguments got attacked, obviously. But guess what? They were actually defended and published in papers on how to refute every argument that came against the critics at that time. So when we emailed Parsons and we asked him, hey, do you want to come on our show and defend your work? He had zero interest. He said, if they want to argue against me now after all these years, they can publish and I'll refute it again like I did in the past. So he's highly confident in these rates. It's not like he just makes them up and just left. Matter of fact, they were confirmed later, which we'll probably get into. So evolution theory estimated one substitution every 300, 600 generations, or one mutation every 9,000 to 12,000 years from a split. Now, that's quite different. Genetics found the exact opposite, with only 24 fixed substitutions in the most diverse people group on Earth, with a fast substitution rate of 1 for every 25 to 33 generations. Completely the opposite. And they even write this in their papers. Look down here and read just this. It is thought that we split from a common ancestor from chimps about 5 to 6 million years ago. More than enough time for substantial genetic differences to develop. Why aren't they there? Where are those differences? Why are we missing millions of years of genetic evidence? Because there's not millions of years. It's really just that simple. There's nothing to confuse or complicate here, right? So with such low genetic diversity, it was not expected, nor the fast rate of change, so it wasn't predicted. Evolution only had one option left, and they needed to invent a global bottleneck in all life to reset genetic diversity. So what could kill all life? Well, there's only one thing, because we're talking about aquatic life too. We're not just talking about land life and mammals and maybe humans. No, it has to kill everything to reset all the genetic diversity. So that is exactly what they did with literally zero physical evidence. They literally managed to dupe the whole world into thinking there was a random global bottleneck about 200 to 300,000 years ago that just reset the genetic diversity. And they went to their own fossil record to look for the evidence, and they couldn't find anything. And it really perplexed them. Well, you can read it right here. He goes, did herring really pass through an equally recent population bottleneck, and anchovies too? You can see they're so confused. Like, why in the world would would like squid, herring, anchovies, fish, uh, rhinoceros, and human beings all go through a global bottleneck that leaves no trace of it ever existing? Well, it makes no sense. And the reason it makes no sense is because it's just not true. It's really just that simple. So this journal study right here says nothing out of the ordinary happened 200 to 100,000 years ago in Africa. What do they mean nothing happened? It had to have happened. It must have happened for humans to come out of nowhere. So again, I don't know why that picture's there. In the following year, 1998, Parsons teamed up with Holland and another team, and they decided to run the mutation rate again, and they confirmed it. They went deeper into the papers just to confirm that the mutation rate wasn't wrong or made up. And they found that there are differences. So they just used the medium. Did you know that the vast majority of the people, the lineages that they tested, 73 of them all had a much faster substitution rate of 1 out of every 17 generations? That's incredible. The slowest rate we got were from Amish people, out of 1 out of an 80. For some reason, the Amish mutate very slowly. They think because they live in the very cold regions and they have children at a much higher age, that's slowing the rate down. But isn't that amazing that there's a variance and that most of them are fast? So can we explain the genetic diversity of all human beings with our model? Absolutely. And it's so easy to do. And it's literally the exact opposite of the evolutionary predictions and exact confirmation of the predictions that come from young Earth creation. And we don't find it just from them. We'll go to the next study, the next following years, and look at this. Both of our studies came to a remarkably similar conclusion, says Howell, whose study was published in 1996. So he got another confirmation in 2003, which was years later after the 1997 and 1998. And then we have another study that comes out that did a deep-rooted pedigree of the European lineages from before. So to match kind of the earlier one I was telling you about with Howell, that got a mitochondrial Eve about 5,000 years ago for all Europeans. Well, guess what? She got the same thing. Hers lands about 220 generations ago to 275 generations ago. That's literally within that timeline. So it just depends on the generation time. So it just more confirms it. Then we have a mathematical program simulation that decided to run the numbers. Let's see what would happen if we didn't test people. But if we were to test what would happen if we just put it in a simulation and let people spread out in the simulation, and how far back would it go until we land on the most recent common ancestor of all people? It landed on the biblical timeline. Exactly what we would expect and exactly what we see in the actual studies. And these are, these studies, our estimations are much higher than the phylogenetic material. That is a common theme that you'll see when you read the evolutionary papers. They constantly reiterate and by saying these phylogenetic mutation rates are much slower than what we observe. So of course, they invoke lots of reasons for that. But these deep-rooted pedigrees right here all give us fast mutation rates. And it's fairly consistent. And it just really shouldn't be. And so we got another one. In 2005, we had another study by Santos, and she ran the mitochondrial D-loop and found 1 out of 29, the same as Parsons. A little bit faster, but it doesn't matter. It's literally within the exact same region that you would expect it to be in from the biblical perspective. Another one, 2012. People don't like that we bring up the Parsons study, by the way. They go, that's old. That's 1997. Why do you guys talk about all the old stuff? It's like we're starting at the beginning, and we're telling you a story about how over the decade after decade after decade, the evidence keeps confirming that it's true. And it shouldn't be. The more they learn in science, the more it should prove evolution, and the exact opposite keeps happening. Why do you think that is? Evolution should be so strong, they shouldn't have their 5 recirculated arguments that they bring up all the time. But that's all they got, dude. And those are dying every day. It's amazing. It's so funny. So again, Why did this guy think their humans are more related? He had to invoke high, massive amounts of inbreeding to keep people more homozygous throughout the years. Well, inbreeding kills off the human population after a certain amount of time, so that doesn't work either. So anyway, all of these are come from the peer-reviewed, published journals that you can find online. They're not young Earth creationist content, so it won't trigger you too bad if you are one to read through it. So again, did humans? Why does YouTube give you this? Because their narrative is to push evolution. They are not young Earth creationists. They are an anti-young Earth creation organization, and you can tell because when you search it, you don't get. Try to search young Earth creation on YouTube sometime. Do you see my channel literally called Young Earth Creation pop up? No, you don't. You see Erica's channels come up first, don't you? That's right. They're against Young Earth Creation. They do not like us. They do not like the content. So don't be surprised why so many critics, complain about this. But then comes Nathaniel Jensen, and now he makes a prediction. He goes, you know what? I'm going to pull the data out of a published study by Ding et al. In 2016, and I'm going to make a prediction that the mutation rate will confirm that we can answer the most mitochondrial Eve, the most recent common ancestor, living about 6,000 years ago. So what he did is he took that and he ran the numbers, and at the high end of the mutation rate that he pulled out of there, the average was 0.16. He used the high end, which was 0.197. He found one mutation every 5 generations. So therefore, the most different, diverse people are 120 differences apart. So if you divide the 5 generations by the mutation, 60, you get 300 generations back. Using a 20-year generation time, you land on Eve 6,000 years ago. Exactly what he said he would be able to do with the material that was given, pulled out from a raw study from the published papers. And that is published in the Answers Research Journal from Answers in Genesis. So in conclusion, studies find that the measured rate of mutations in human populations using pedigrees have reported values that differ significantly from rates estimated from the phylogenetic comparison of humans and chimpanzees. And the reason that they're always different is because the one is made up and not true, and the other one is. And when you look at charts based on evolutionary assumptions, the estimated rates down here for evolution never show up. Not one time does anything ever fall within this region. They all fall up here in the observed very fast rate. What does that tell you? Evolution is not true. It really is just that simple. So we can come back. I'll just close. I'll stop sharing.
Share.
So
yeah. Thank you. I appreciate that. That was awesome. And I have a question on the mitochondria, even the mutation rates and stuff. Do they have a response to that? Have you heard critics give any kind of response? Not a good response, just a common one maybe that critics, or even a couple common ones that they might be thrown out there. Have you heard any?
yes, I have. They've got a list of them actually. I like to go through their list, but I will show you the list if I can find it. I didn't think we would have time for the list, so we'll look at them real quick. The
We do
first
now,
one that comes to mind. Yeah,
we
that's
do
true.
now.
Yeah, that's right. Okay, the first one that comes to my mind is you guys realize that the most recent common ancestor doesn't refer to Eve, right? It's just the most recent common ancestor. I say, okay, fine. So what? What bottleneck 6,000 years ago in evolutionary model could possibly account for every human on Earth not having male or female children? Are you kidding me? You think that that's going to bottleneck the whole world down that just so happens to land on the biblical timeline?
Don't they
No way.
literally call it mitochondria Eve?
They do. They call her mitochondrial to be a mocking slap in the face to young Earth creationists. They did that to mock us, actually, which is ironic that it turned around and bit them because they used it to mock us and then it actually ended up being true. So here's a list of their arguments. Studies that probably counted somatic mutations. What somatic mutations are there mutations that build up inside of us as we're alive right now. Like let's say we love to smoke and drink. We're going to get a lot of mutations that come from that, but they're not going to be passed on to our children. They won't be inherited. So if you count them, you're actually going to speed the mutation rate up. It'll be by accident, but it'll look really, really fast because of it. That was their first argument. The next one is they said, you guys didn't consider coalescence. That's a kind of a slap in the face of all the scientists that literally did a substitution rate, like Parsons and Howell, who literally did a coalescence calculation based on the maximum diversity of human population. So yes, they did. The next one would be that the substitution rate is slower than the mutation rate. Okay, so granted being a little bit slower, can we still answer it in the biblical young earth creation model? Absolutely, because not only do we have one bottleneck with the global flood, we have two bottlenecks because we have the Tower of Babel bottleneck that comes afterwards at all. So we have two bottleneck where the human population gets very small, which makes mutations raise to fixation rapidly fast. So of course we can explain why the substitution rate is fast and why there's so few substitution, even though that rate is fast. Another one, this is their favorite, is purifying selection probably removes mutations over time. So when you're looking at mutations building up after 1 or 2 or 3 generations, sure, that looks really fast. But if you go back 20 generations, well, guess what? Maybe those mutations will just disappear and you won't see them anymore. So what I say to that is something really simple. Let's go and see if that's true. Well, guess what? They did do a study on that, a deep-rooted study on chickens, and it went back 50 generations. And when they traced all 50 generations of 2 separate lineages going all the way back, they found that, again, the rate was 15 times faster, right here, 15 times faster than the phylogenetic mutation rate, exactly what we would expect, and substantially higher than the calibrated fossil rates. Surprise, surprise. So what do we find again? We find the exact same thing in deep-rooted pedigree studies. The very thing that they said you wouldn't find it in, well, we find it. So that rescue device is also dead in the water. We can go over other studies too, but I love that one just because it's so simple and just shows how desperate they are for these not to be true. And then another good one would be, well, the mutations still point to Africans as being the oldest population because they're the most diverse. It's like, well, of course they're the most diverse. They have no agriculture there. You don't, like 100 years ago, you go to Africa, they don't have farmland. They don't grow, they don't have massive civilizations in there. So they don't have large populations. You go into South Africa and they found that they lived in tribes in small populations where it's very hot for their whole history. So what do you expect to find? You would expect to find more mutations in small populations. It's exactly what we do find. So it's all so easy to answer. The more a population spreads and the bigger it gets, the better selection works at removing mutations. That's why the bigger the population you get, like over here in America, you don't get a lot. We're never going to reach fixation now. People are mating with a population size of hundreds of millions of people, right? There's no way you're going to reach fixation anymore. It can only happen in small populations. So that's what we see when we look at smaller tribes and we go to Africa. They have faster mutation rates and they have more fixed mutations. It just makes sense. It's so easy to answer from our model. And it's not because they're older, they're all age. And just like today, let's say they were to test me versus some guy that's Amish. Well, in 5 generations from now, the Amish generation is going to look like they're much younger than my generation that's going to look like it's much older. And it's just not true. We're exactly the same. So it's just very easy to answer.
Right. It's interesting that you mentioned the African one because me and Dr. Peter Lyon next week will be talking about the so-called out of Africa theory that they believe in, that we originated from Africa. So he's going to be offering a response to that. So so if I'm understanding it correctly, because the people in Africa was far more isolated for a long time in a particular region of the world, you say that's the reason why they have more diversity or less or what?
That's the reason they would have more, but there's also other reasons as well. So when you look at a genetic chart, you see over here that this is the longest branch of a tree, meaning that line has more mutations than the rest of all of the other lines on Earth. So African branches are very long, meaning they have a lot more mutations. So if they have more mutations, evolution sees that and says, well, they must be much older. We say no. When you look at the different mutation rates in different people groups, some are faster, some are slower. And when we look at Africans, they actually have more variants of a PRDM9 that are rare in Europeans, which is a recombination point. So they have faster rates of recombination, aka more mutations occurring inside of them. So that's another reason why they mutate fast. So not only do they, what is this? Africans generally exhibit a higher rate of mutation compared to other populations. Surprise, surprise. So if they have a faster mutation rate, if they have more PRDM9 sites, if they lived in smaller populations, if they live where it's warmer, all of these things contribute to there being a faster mutation rate. If they have a younger generation time, that would also mean more mutations over the exact same amount of time. You know, if one lineage like me and goes 10 generations, but we all wait till we're 40 to have kids. Well, guess what? An African that has kids at 15, they're going to have double that amount of populations after that same amount of time. So they're going to accumulate a lot more mutations than my line will. So it's amazing. So there's a lot of ways to answer why Africans have so many more.
What would you say on China? Because they often are said to have a population control issue, and they have a lot of people and that exist in China. So how would you, what would your explanation be on the mutation rate concerning China as far as that go? Do they have a lot of
China does
diversity? Do
have
they
a
have
very
less?
low.
Go ahead.
Well, China does have a large population, but they also are government-controlled communists, so they don't allow a lot of travel there and out. So they have, though they do expand, that's great. Once that expansion stops and they kind of have a population neutral, you know, where it just kind of hangs out at a net zero population growth, that's when the problems come. Because the population needs to continue to grow. And if a population stagnates or starts to drop, then you can see a little bit more of a problem. And you're right. In China, they're not as diverse as Americans, where our borders are more open and people can come and fly around. But they also don't have too much of a problem where they have to worry about inbreeding either, because their population is so big. There isn't like a worry of like, oh wow, their mutation rate is just so fast. Now it's pretty common it's pretty around the rate, much as the same as us.
Right. And while we're on the subject on that, you have a particular argument on our phylogenetic diversity or similarity or diversity, the way we are to each other versus the way apes are to each other. Um, and you have a particular argument on that that was kind of interesting if you wanted to talk about that.
Sure. Let me just pull it up. I got a lot of folders right here. And yeah, I like that topic a lot because phylogeny is
Yeah.
one of their favorite arguments now. It comes down to like saying, well, the fossil record is true and we can see these small changes. And then when we look in the genetics, guess what? We see the same thing. And that proves that there's this perfect line of connected diversity of ape to man. It's kind of like, no, you have to do so many mental gymnastics to get that to work at all, that the second you take away any of that myth and you compare other regions, it's game over. Kind of like I did on that radiometric dating video recently, right? The second you compare other methods to each other, it's game over. The same thing in the human genome. If you line up the other regions like cytochrome C with cytochrome B, they don't match. And so if they don't match, then there is no phylogeny. And here's a perfect example. Here we go. Let me share my screen again. This is the chart that comes from the cytochrome C. This is the highest conserved regions that they build their best mutation, their phylogenetic trees from. They don't build it from cytochrome C. They don't build it from superoxide dismutase. They don't build it from blood samples. No, what they're doing is they're looking in the mitochondria and they're looking for the most conserved region, which is cytochrome C. And they say, look at the tree. Look at this tree. It's a perfect nested hierarchical tree. It's actually not. After just 3-4 steps, it's already broken down. Actually, I think pig is even before gorilla. But let's just go with that. Look at this. We got human Neanderthal being completely related, identical. And then chimpanzee pops up. Okay, we explain that through genetic differences, but we'll get into that. You're thinking, okay, that's a pretty good line. But then it's not the orangutan. The pig pops up, but why? The pig has 98% similarity. That's really high. That's more than the orangutan. And they admit this. And then after the orangutan, guess what? There's a dog. I don't know why it's not in my picture here. This must be a really old one. So we have chimpanzee, gorilla, pig, orangutan, and then dog. The phylogenetic tree of human ancestry. I don't care what anyone says. Matter of fact, this was so perplexing that a geneticist looked at this and said, wow, I don't understand what I'm seeing right now. Look at all these differences. Pigs have a lot more similarity to humans in these regions than chimpanzees do. Now, what does the young earth creation model say? We say that genetic similarity isn't based on relation, it's based on function. So if we share a kidney that's more similar with a pig, that sequence in that cytochrome C will be similar to the pig and not the chimpanzee. And that's why we'll share more similarities with certain things and then not others. And that's exactly what we see. That is why when you look at the eyes, we have eyes way more similar to a pig. Did you know if we were to remove blood from a pig, I think it's called xenotransmorphia. That's how we transport. We can use pig blood into us. If we were to use it from a chimp, it would kill us. Pig blood is more compatible than a chimpanzee. Supposedly, our most recent common ancestor by millions of years closer than a pig. So what's going on? Well, he's this Eugene McCarthy. He said, well, there must have been a pig chimpanzee mating hybrid that created humans. And this is a real thing. This is a literal thing that exists. But we still have to go back in time and say, well, look at this. What does this chart show humans are related to? Well, it doesn't look very good, does it? When you compare cytochrome uh, C, which shows what we just talked about, humans, Neanderthal, chimpanzees, gorilla, pigs, pongo, and wolves and dogs, well, guess what? That's already not good, but that's the best they've got. When you compare cytochrome B, well, guess what? Now you have humans nesting with whales and cheetahs and lemurs. What? And then a monkey. What the heck is going on? Where's the phylogeny? Which one is our cousin and why? Which one are we more related to? Why are we more related to a cheetah here than a chimp? See, phylogeny breaks down when you look at the phylogeny in a different region. So they pick and choose. They cherry pick a region to look at and ignore everything else. They don't even compare them to try to build a better one. And their excuse for this, you'll like this. Well, why is it like that? Well, mutations randomly mess that region up. We can't trust it. Oh, well, what about this? Oh, well, mutations must have messed up in that region as well. Oh, so we can't trust it. The very thing that you said we can trust to prove lineages go back in time, you're now telling us that we can't trust it because mutations can mess it up sometimes. Oh, this is real great. I love so anything can be anything, basically, and it's still good evidence for evolution. It's just buffoonery on every level. I just can't get around it. It's so ridiculous.
Yes, yes, absolutely. It always interests me that see because if you actually sometimes people talk about chimps and humans being 96 to 98% similarity. Then of course with these algorithms they only look for similarities. They only look for alignments, and so they don't really count the differences too. And then it's like you said, it's really the type of similarities and differences. We look for functionality. Sometimes like certain types of organisms will have similar needs and that sort of thing. And there is one thing that did pop in my head while you were talking on that. There are some people that say that chimps are born without what was it? Uh, I want to say Lycine. I was like, no, that's Jurassic Park. They're like born like we both don't have we have uh being able to adapt to what was it? Ship sailors had this issue for a long time, and they realized, oh, we don't have. I think it was vitamin
Scurvy.
C.
Scurvy. It's
Yeah,
it's a broken gene that doesn't allow us to absorb vitamin C, or
C.
make
Yeah,
vitamin C in our bodies
that's
anymore.
it. That's
So
it.
it's a broken, yeah, it's a broken gene. It's like saying, well, my car, every time I drive it, my tires get messed up. And because they get messed up, this must just be, we must be related. That car must be related to that van. It's like, no, some areas are weaker than others. Just because it's broken in that region doesn't mean that it's shared. It just means that we shared the gene and that they probably broke in the same area. That's just exactly what would happen with a region that's weak. There's certain strong areas and there's certain weak ones. We tend to mutate in the same regions. Mutations congregate in certain areas and they don't congregate much in others, meaning there's some conserved and there's some not so conserved. So it doesn't really matter how much mutations accrue there. Other areas are weak, some are very strong. So humans were created to be fruitarians. And if you look at a chimpanzee, guess what? They can eat all the plants in the world. They can go over to a leaf and rip it off a plant and eat it. You try that. I dare you. You think you're related to that monkey? Go ahead, go eat that leaf. You're going to realize real quick you cannot do that. Well, why? Like, what's going on? Well, our appendix was never, ever what a chimpanzee's was. Meaning they say our appendix is vestigial. It lost its primary function. What is its primary function? Well, if it's related to a chimpanzee, it would have been to only break down plant cellulose. That's it. A chimpanzee's appendix does nothing else. Guess what a human's does? It's an immune system. It produces killer T cells. It produces killer B cells. Killer T cells work on the outside of the cell. Your thyroid makes them. Your killer B cells, which are produced mostly by the appendix, work on the inside of a cell. So it's a backup immune system. It also harbors good bacteria to push out into the system when there's an infection. It has all kinds of functions, and none of them have anything to do with breaking down plant cellulose at all. They never have. They've never even found a human being with a mutation that can break down plant cellulose. The only reason we can eat plants is we cook them, or we blend them, or the hydrochloric acid of our stomach can digest them. They can break them down. Without that, we're in big trouble trying to eat greens. That's why you have to juice them, blend them, steam them, ferment them. Trying to eat them raw is very hard in the digestive tract because we're not apes. It's really just that simple. But we were fruitarian. The Bible says we were created, placed in a garden, and told to eat fruit of the tree. And that's exactly what we are. That's why our teeth are shaped like fruitarian. That's why our mouth produces amylase and our tongue is carbivoric, meaning we are designed to eat fruit, which is a carb. And that's why everything about us is designed to break those things down, not to break down green or meats. Without the hydrochloric acid of our stomachs being that way, we couldn't do it at all. That's why humans cook their food, because 90% of the foods we eat today, we can't digest without cooking it to begin with. So yeah, because they can't digest or produce vitamin C like we can. So what? I like to point out the things that we are different. Everyone tries to look at the similarity. You want to look at a list of how different we are? We could go through 30 minutes straight. I have a folder right here. It is going to blow your mind if we went through that.
Right. And they when Richard Buggs did that analysis of us and Tompkins, and they were 85% similar, and the 50% difference actually amounted quite a bit in terms of the amount of differences. So and it's not just the amount of differences; it's also the kinds of differences that we would have from chimps. That's significant. A lot of people don't really think of that when they think of those issues. Um, and I'm kind of interested in the mutation rates. I know Donnie isn't here. Usually he shows a graph from Dr. Nathaniel Jensen, and I know you mentioned him as well. That he had, I don't know, maybe you have that graph too. I have it. I actually
Yeah.
have it up on mine too. Unless you
Oh,
want to go ahead
good.
and show yours. Well, I was going to sit there and
No, go
look
right
at.
ahead.
Yeah, here it is. Three haplogroups of mitochondrial DNA, actually. So that fits our subject pretty well. Do you want to explain these three haplogroups real quick?
Okay, yes. Yeah, this is a chart looking at all of the different mitochondrial lineages in women from around the world. And you'll see that the green arrows in the middle are pointing to different of the three branches that populated all of those people group. So we have the one at the very top, and a matter of fact, let's start at the one in the middle. If we follow that to the right, that would lead to the lineages of the African population, which biblically would be the line of Ham. And those are the ones that have the most mutations in them. And if you can see and follow it to the far right, you'll see it says sand individual. That is a group of African population people that have the most genetic diversity on Earth.
Am I
So
just to
therefore, they
make
are
sure?
considered the oldest. Yes,
Just to make sure I got it right.
that's it.
Is this right? Okay.
That's right. You're exactly on them. And that individual that was tested had about 120 mutations from that green arrow there in the middle. So the L, M, and M have the least amount of mutations, but they do have a mutation differences between them, though not very many. Matter of fact, one has so few compared to the other arrow that they're considered to even be sisters. So very few mutation differences between L, M, and M. And then from those branches, the whole world population flows from those three. Nathaniel Jensen chose those because of the predictions that come from the Bible saying that three females had three different types of mutations, got off the ark, and then repopulated the world. And then from the Tower of Babel, they would branch off from that yet again. So this is a biblical map of ancestry that we're looking at.
If I understand correctly from Creation Today, you mentioned and referenced those three as the three daughter-in-laws of Noah. Is that right?
That's right. That's exactly right. The three daughter-in-law of Noah. Yep. And they would go genetically, they would just go by L, M, and M. Unlike mitochondrial Eve, because you see, we can track back to these three progenitors of all these people groups, but we can actually go beyond them altogether to a single point as well, which would take us all the way back to a single most recent common ancestor. So these L, M, and M are the three nodes, but that Eve would actually land somewhere on this branch to your right, somewhere near that first blue arrow. That would be
Okay,
the
so
root of this tree. Actually, the one right in to the left of that. Yeah. So follow that down, count over about three arrows, and you're somewhere near the L node root for this tree. That's where Jensen actually placed Eve. So very interesting.
oh,
Yeah, you're
awesome,
pointing at those
actually.
arrows.
Yeah, I'm
Yep.
not
So
exactly sure
that,
which if I'm even playing the right arrow.
yeah, or at least you're in the right area, so you're doing good.
Somewhere in the vicinity.
Yeah, yeah.
All right, I'm gonna go ahead and
Eve,
stop
Eve somewhere
sharing.
on there on that chart. Yeah.
Right, right. I'm gonna go ahead and stop sharing.
Okay.
All right, awesome. Make sure everything's recorded still. All right. Yes, that's pretty cool actually. I thought that was pretty awesome when I saw that y'all put that on Christian Today, and you talked about mitochondria or those three haplogroups, and so I thought that was pretty cool because it does actually look like that. I mean, from a biblical creationist perspective of a global flood, Tower of Babel, create Eve as the mother of all living, bottleneck at the flood. I mean, when we start uh, zooming in and looking at this evidence in light of what the Bible says, it's looking extremely like the biblical text is correct. Am I right on that?
Yeah, exactly. That's exactly what the Bible predicted we would find, and that is exactly what we did find. Now, of course, evolutionists have rescue devices, but so what? We could have rescue devices for anything as well, and the second we come up with them, okay, God of the gaps, God of the gaps. It's like you guys use evolution of the gaps literally every day. So I don't know what they're complaining about. They just like to complain. That's all I've ever come to determine at this point. I've been a young Earth creationist, and I've never met a more vitriol group of people in my life. And just think, I was part of a community. I've been around vegans. I've been around groups of people that will like burn cities to the ground, and they're nowhere in comparison to how bad atheists are. Not even close.
Actually,
Man.
a thought actually crossed my mind just now. During your presentation, you mentioned that evolutionists get all offended when they're called evolutionists, and you like, well, I don't get offended if I get called a creationist. I said those exact words to a critic one time. It's like,
No
why do
way.
y'all get so, yeah, I said, why do y'all get offended? Us calling you a evolutionist. It makes no sense to me. I mean, what's the deal? I think they're afraid that people will realize that what they're holding to is a belief, and they don't want people to think it's a belief. They think that they want people to think it's fact, and it's not, and that sort of thing. And so they get uncomfortable when you reference their position as a belief system. Now there are evolutionists. I've met evolutionists that actually admit that it's part of their belief system. And there's some evolutionists that
Nice.
admit they have a worldview and everything else. And then there's others that will deny that, like they got fire in their veins. They do not want to admit to having a belief. And maybe that makes them think that you're accusing them of that, which I don't need that to accuse them of. I accuse them of it anyhow. But evolutionists is just what they are because they believe in evolution. And they argue for evolution, and that's the position. Like my position is creationism, and I hold that proudly. I don't, of course, if I was an evolutionist, I'd probably be embarrassed to be called an evolutionist too, but
Yeah. Yeah, I wish somebody had called me an evolutionist when I believed in it. I'd be like, "Oh wow, I never knew I was classified as one." It's just like one of those things that you just believe it because you were taught it in school. You didn't question it. You didn't consider like, "Oh man, this I'm an evolutionist now." Like it's a label. And the fact that they're ashamed of it tells me everything I need to know. That's all I needed to know. Oh, you're ashamed of your label. Oh, thank you for admitting that. I now I know you don't like that you're related to the strawberry you ate for breakfast. Okay.
yeah, they don't like us referencing that either. Because they think that we're related to plants, but that is their position. I mean, it's we're not making fun of them, of course. We do find it funny, but we're not
That's
trying to make
true.
fun of them. That is their position. It's not a straw man argument. It is what they believe. It
It
is what they
absolutely
argue.
is.
And the funny, and I always thought it was funny that evolutionists will laugh at us because we believe in creation, we believe in young Earth, we believe in a global flood, and then they would find it. I don't know, some reason they find it hilariously funny, and I'm like, okay, do you realize that if I wanted to laugh at you guys, y'all make it so easy for me. Y'all believe that we're related to chimps. Y'all believe we're related to bananas. Y'all believe that the whole universe started with a dot. So
Yeah.
y'all
I
make it
like when they
really easy
say,
to make fun of y'all. So but I don't
yeah.
do that because I do actually try to be serious about your arguments and deal with that and respond because there are people that actually believe that, and I think that evolution is a very dangerous perspective. Which brings me up another thing. And the reason why I was thinking about that just now was because way you mentioned their evolutionist attitude, the way they're dealing with these and responding to these things. And often I tell people like, I don't usually start out with accusations of them having a religion or whatnot, but they do act as if they have a religion, regardless what they might say about it. They are extremely dogmatic about it, and I think evolution. This might be an interview wouldn't mind having with Dr. Bergman, but I think evolution actually hinders science. It prevents the progression, because science is supposed to be held provisionally. You're supposed to be able to advance in science. You're supposed to be able to update your knowledge in science, and what you think you might know one minute may not be what you might know the next. But it's like every time that we find something out, instead of changing their views, they hold on to it and they look for rescue devices and things like that. You know what I'm saying? And
Yeah, absolutely. That would be a fun talk too. I love going on that one because you're absolutely right. So right.
and that would be a serious problem. With because I think it does. I think acts as a science hinderer. It tends to hinder people in actually practicing real science. And I had someone I actually come up with my own model. I know you have one too, but I have my own model in the so-called heat problem. And I had a critic that came and say, "Well, nobody else came up with that idea until you came along with some of the things that I've come up with are suggestions that have actually been made by the creationists. I just modeled it.
Yeah.
But um, but it's interesting thing is like, okay, isn't science supposed to be about updating your knowledge and growing in your thinking and thinking outside the box and with different people having different ideas, but y'all make it look like science is this dogmatic concept that you just cling on to that one perception you had, that one view, it's the only right view, and then if something else contradicts that or some other data contradicts that, they come up with some sort of rescue device to try to explain it. You know what I'm saying?
Yeah, it's so common. That's what they literally do is they go, well, evolution is a fact. So what else can we know from that? It's like, wait, you've already made up your mind. That's exactly the opposite. You've already put yourself in an unfalsifiable dogma position, and now we're trying to prove everything off of that. It's crazy.
All right. I will talk to you later, Matt, and thank you for coming on. Thank you for agreeing with this. I've always wanted to interview you, but you usually don't have that much time to engage in interviews. So I'm glad that you were able to give me some of your time today.
Yeah, absolutely, man. It's real hard with kids. It's hard enough to record audios. Oh my gosh. I mean, up all night trying to do things like that, but as you know. But yeah, thanks for having me on. We'll do it again.
Awesome. All right. Appreciate
I
it,
can't get it.
and I'll see you later. And God bless.
All right, man. Thank you. God bless.
I decided to add this explanatory comment in the middle between Matt's and Donnie's interview. As I said before, I had to interview Donnie separately because of a mistake that he had made on the dating, but everything worked out great. And so we continued to have our interview, and so I decided to splice everything together right here in the editing and show you Donnie's interview. And I think Donnie's interview and Matt's interview really complement each other. Matt talked about the haplogroups. Donnie also discussed that. So it really complement each other, and combining the two, I do have a really really thorough discussion here between Matt and Donnie concerning the genetic evidence that confirms the Bible, and so that's what I was really going for in this video. I know it makes the video kind of long, but it's okay. There's pretty much everything that can be discussed on genetics that confirms the Bible in this video. So I'm really happy about the results. So without further ado, let's dive into Donnie's interview with me. Hello and welcome to Apologetics 101. I am here today with Donny Budinsky from Standing for Truth Ministries. I know y'all probably got through seeing an interview with Matt from Standing for Truth. So we're going to be delving a lot more deeper into these issues concerning genetics. And we're going to see quickly how well genetics actually confirms the Bible in today's video. And Donnie, do you want to I know that you've been on my channel several times, but do you want to real quickly introduce yourself and your ministry, and then we can jump right into it?
Brian, as always, it is a pleasure to be here with you. My brother, you're doing excellent work upholding the truth of biblical creation. So God bless you for that. And I appreciate another invite here and your willingness to be seen with me in public for another couple hours. So, Brian, all jokes aside, this is great. My name is Donny Budinsky. I do run the Standing for Truth Ministries, where we focus on all things biblical creation and origins.
Oh, awesome. I'm glad to have you here, actually. And today we are going to be doing my big genetics video from my Are We Apes series, and I can't wait till we jump in. We're gonna be dealt. I know we dealt with some very specific issues in the past genetically. Talked about Y chromosome and the chromosome 2 fusion
Chromosome
argument
2
that
fusion,
in the past, and of
ERVs.
course ERVs. Yeah, I was fixing to say that. ERVs. So we delve in those specifics. So if anyone wants to like get a more in-depth approach to the particular issues that we might be talking about today, they can go to those videos, and we go really deep into those particular subject matters. But today
Yeah,
it's
if
kind of
I could,
like,
Brian,
yeah,
I think with the fusion, our two shows combined are roughly 5 hours or so. I mean, there's a lot of content there. We left no stone unturned. We dedicated an entire program to addressing the critics, refuting their objections. And then we also did a program responding point by point to Gutsick Gibbon on junk DNA. And so the ball is in the court of the evolutionary community to counter those arguments, brother. And so I appreciate your willingness to be comprehensive on this topic, which is something I think we have to do.
right. And usually, most critics won't even touch junk DNA claim because
Mm.
I mean, a lot of internet Well, I guess maybe we'll get into that. I don't know if we're going to be covering. Are you going to be covering anything on junk DNA today or not, or is maybe
I
briefly?
could probably touch on it for a minute or two. And, it's pretty easy to show that junk
Right.
DNA is false. So I could probably go over a couple arguments there. And I will say for those that might have a question or maybe 20 questions on origins, if they go to the website, I've got my website fully updated, Brian, standingfortruthministries.com. And if you go to the answering challenges section or the video section, you'll find answers to just about everything in the world of biology, genetics, geology, astronomy. And so people, feel free. I encourage you to utilize the website. If you go to the video section and scroll to the bottom, there's a section that says guests on Standing for Truth, and you can see a list of a lot of the scholars and PhDs that we've had on, specializing in different fields as well, interviewing a lot of the same people that you've interviewed. So I encourage people to check that out.
Oh yeah, I would definitely encourage that as well. And of course, I need to update my website. I'll probably be doing that after March, but I need to update that and renew it and everything else. So I'm hoping that I can be able to do that eventually. A lot of great content. You produced a lot of guests on your channel, and you have them on a regular basis. And I would implore anyone to subscribe to your channel and to see your content. Go to your website. Um, your website's usually it will be linked in the description box of this video as well, but it's usually linked in your videos too. Correct to your in your
Yeah.
comment or discussion descriptions. All right,
Yeah.
awesome. All right, without further ado, do you want to jump in? You have a presentation to present us. Is that right?
I do. And I'm excited to show this. I've been working on this presentation, making sure that we are leaving no stone unturned and utilizing the best possible arguments. Brian, you're going to love this because you and we've done a lot of behind the scenes work on this specific topic of the Y chromosome dissimilarity between humans and the great apes. And so I will share screen. So, Brian, let's get into our points here. And what I want to do, though, here's a sneak peek. We are going to be discussing the science of separate ancestry. And I like to focus on what's called the differentiating evidence. Now, what I mean by that is there are a lot of arguments and points of dispute in the creation versus evolution debate that are largely agnostic to the overall debate. What I mean by that is both models can generally account for the data. For example, if I were to say, well, the sky is blue, therefore creation is true, the evolutionist would say, well, that's a little ridiculous because we can also explain why the sky is blue or the earth is round. So there's a lot of overlap in the creation versus evolution debate. Let's pretend what we see in the Marvel universe, the multiverse. Let's pretend that's true, Brian. So let's say you got Earth 2 and Earth 3. On Earth 2, creation is true, let's say, okay? Now, even though creation's true on Earth 2, we could look to a lot of pieces of evidence that even though evolution is false on Earth 2, the evolutionary model can still explain. Okay. Let's say on Earth 3, evolution's true. Well, you'd still have a lot of pieces of evidence that even though evolution's true on Earth 3, the creation model can also explain. Okay, that's why a lot of arguments, let's say in the world of homology, nested hierarchies, even these interesting mosaics like transitional forms that we see, they're largely agnostic because both models have good explanations, right? Homologous structures, the shared structures that we see in the biological world. Well, creationists would look to those shared structures as being the result of common design, and the common descent position would look to homologous patterns in the biological world as being the result of descent with modification. So point is, both models can mostly explain the data. Now, I would say once we get into the real details of it, like with homology, I mean, even in similar sequences now between humans and chimpanzees, they're finding significant differences in the way those genes are regulated, the way they're used, the way they're expressed. So once we dig deep, even on a lot of these lines of evidence that appear to be agnostic, you start to realize that creation has the far better explanation. Okay, so all of that to set the foundation. We're going to be focusing here today on the differentiating lines of evidence, those pieces of evidence that can discriminate between the models. As in, here's a data point, here's a piece of evidence that only creation can explain and actually precludes the evolutionary model. And one of those is going to be the Y chromosome, which I know, Brian, you've also studied a lot on. So before we get into the science, though, I did want to cover a couple scripture and just show everybody why the separate ancestry model is exactly what we see in the Bible. Okay, so let's go over some of these scriptural points. What does the Bible say? Well, the book of Genesis makes several genetic statements with significant implications. And I want to go over some of those statements. The Bible claims there's no common ancestry between humans and any other form of life. You're going to see that. What does the Bible say? The Bible claims to be what? The history book of the universe. Okay, the Bible describes the creation of two people, Adam and Eve, just 6,000 years ago when you account for the genealogies and the timeline provided in the scriptures. Jesus says from the beginning of creation, he made them male and female, referring back to the creation week, referring back to the creation of Adam and Eve. The Bible calls Adam the first man, Eve the mother of all living. And we see no hint of ancestry with the great apes, dogs, bacteria, fish. Notice this, Genesis 2:7. And the Lord God formed man out of what? The dust of the ground. Do we see Adam? Do we see humans coming about through evolutionary processes, evolving from pre-human hominin-like ancestors? No. God specially creates man out of the dust of the ground. And the rib which the Lord God had taken from man made he a woman. So Eve, created from the rib, specially and separate. When we say separate ancestry, people might be wondering, well, what does that mean? That means that humans are created separate from any other form of life in the biological world, rather than the opposing view, which is universal common ancestry, right, Brian? That says all life, including plants, including animals, are related through common ancestry. Now, we shouldn't be surprised, Brian. I think it's important to point out we shouldn't be surprised when critics and skeptics, they will be presented with this evidence, this truly incredible evidence for special creation. And yet, what do? They go running to rescue devices, rescue mechanisms. They deny the data. They scoff. Well, this was predicted, brother. 2 Peter 3. Knowing this first, that there shall come in the last days scoffers. We've been in the last days for a couple thousand years, walking after their own lusts and saying, where is the promise of his coming? For since the fathers fell asleep, all things continue as they were from the beginning of the creation. Brian, what does that sound like? Uniformitarianism, or actualism as they call it now? For this, they willingly are ignorant, that by the word of God the heavens were of old, and the earth standing out of the water and in the water, whereby the world that then was, what is this? The global flood, being overflowed with water, perished. And the global flood has such incredible explanatory power in terms of what we see today, the geologic features of the earth, the fossils, the rapid formation of a lot of things. Okay, limestone being one of them, caves, for example. And so when you deny the Genesis flood, you deny an elegant explanation for a lot of the observations on the planet that we have in real time. And so the scoffers here are denying what? They're denying the special creation, right? They're denying the flood, and they're denying the coming judgment. But the heavens and the earth, which are now by the same word, are kept in store, reserved unto fire against the day of judgment. So 3 things, and these are the 3 things that we debate the skeptics with. So don't be surprised when you come across critics that deny the evidence, reject the evidence, because some people are just unfortunately willingly ignorant. And okay, let's continue here. So we have significant implications if we were to deny the clear account of origins in Genesis. If there was no first Adam, then what is the necessity of the last Adam? If Adam did not really exist, then why did Jesus die for the sins of the world? Notice Jesus in Matthew 19, for our Lord and Savior, God manifest in the flesh. In the beginning was the Word, and the Word was with God, and the Word was God. And the Word became flesh and dwelt among us. That's God manifested in the flesh. That's amazing. Okay, and what did he say? And he answered and said unto them, have ye not read that he which made them at the beginning? So going back to the passages we read earlier in Genesis, Adam was made from the dust of the ground, Eve from his rib. They were created special. They were created separate from any other form of life. And Jesus says this all occurred at the beginning. This is creation week, day 6, to be specific, Adam and Eve. Okay, we see no hint at universal ancestry. And he answered and said unto them, again, have you not read that he which made them at the beginning? Mark 10:6, but from the beginning of creation, God made them male and female. 1 Corinthians 15:45, the first man, Adam, he was the first human being. Eve, the mother of all living. You see how this all comes together beautifully? For in 6 days the Lord made heaven and earth, the sea, and all that in them is. Notice that all. This is emphasizing everything, including humans, Adam and Eve. And rested the 7th day. Wherefore the Lord blessed the Sabbath day and he hallowed it. This isn't about, hey, what does this really say? Let's debate this. No. The question is, do you believe what this says? Because it's clear. Genesis 5:2, male and female created he them. Again, Mark 10:6, Genesis 3:20. And so you can go through all of these many amazing passages that make it clear, Brian, that Adam and Eve were the first 2 human beings, and they were created separate from chimpanzees, orangutans, and so on. Right here would be a representation of the universal common ancestry model, which should suggest all life. Notice here, this is a sophisticated phylogenetic tree of life, okay, where you've got everything, including bacteria, eukaryotes, fish, amphibians, mammals, right? Think of the evolutionary story as what? Farm, fish, amphibians, reptiles, mammals, and eventually man. That's the evolutionary process, large scale evolution. So they believe everything can be traced back to a single common ancestor. Whereas the separate ancestry model here, this is the model we uphold and defend. You see on one side, you've got humans. You've also got the extinct human groups, fully human, okay, after Babel. Some groups are even before. They dispersed, some became isolated in different parts of the globe. And a presentation for another day would be the details on that. Neanderthals, Hobbit, Denisovans, so on and so forth. Okay. And so this is where I want to get now into the scientific evidence that confirms exactly what the Bible says. Brian, my brother, you saw it. You in the audience, you saw it. What did the Bible say? Well, it did not say anything about evolution. It did not say anything about common descent. It clearly taught separate ancestry. Now, you got theistic evolutionists that come along, okay, and they want to reinterpret it, over allegorize it. They want to try and twist what it's clearly saying, right? But we don't have to do that. We can believe what it clearly says, and what it clearly says and teaches is separate ancestry. And it just so turns out that the scientific data confirms that, confirms these genetic statements with significant implications. Here are some predictions. Low genetic diversity, that's what we see. One Y chromosomal ancestor, that's exactly what we see. Every single male Y chromosome on the planet can be traced back to a single male ancestor just 4,500 years ago based on the known mutation rate. It's fast, somewhere between 1 and 3 per generation. There's low diversity in the Y chromosome, in the mitochondrial DNA, low diversity in general. Okay, I talked about this on Creation Today about a month or so ago. And it's a truly fascinating data point, because if God created, Brian, let's think about this. If God created Adam and Eve just 6,000 years ago, what would we predict? Well, we would predict today that humans should have low genetic diversity. Why? Well, God makes it clear in Genesis that he creates animals in bulk, in fully working and functioning ecosystems. Okay, yeah, he creates humans in two, but he doesn't just create two felids or two canids or two fish. No, he creates populations of animals. So we would actually expect higher diversity in animals, lower diversity in human beings. Okay, because starting off with two restricts the amounts of genetic diversity. The evolutionary model, on the other hand, notice this. They have millions of years of evolutionary history and forward evolutionary change, right? Again, you go fish, amphibians, reptiles, mammals, eventually apes. Then you have Australopithecine-like ancestors, right? After the split, where humans and chimpanzees supposedly go back to a common ancestor 6 to 9 million years ago, you have a split. In the human line or lineage, you have hominins, okay? And those hominins include what? Australopithecines, Homo habilis, Homo erectus, Homo neanderthalensis, so on and so forth. So what does the evolutionary model have? Well, you got the Australopithecines. Mutations add something, Brian, because they're adding something that was not previously there. So they add genetic diversity to the gene pool. So if you have millions of years, or even a couple 100,000 years, if you want to go to their bottleneck, of mutations accumulating every generation, you're adding genetic diversity to the gene pool. So that's a lot of genes floating around these populations, from Australopithecines to Homo habilis to Homo erectus. The story says Homo erectus lives on the planet for about 1 million years, leaves Africa. Some say evolves independently into other hominins like Heidelbergensis, Neanderthalensis, Homo floresiensis on the island of Flores. In Africa, they continue to evolve eventually into humans. And then humans spread out into all parts of the planet, seizing dominion over the planet, and also meet up with other hominins like Neanderthals, Denisovans. So the point is, we would expect higher levels of genetic diversity in all DNA compartments. Autosomal DNA, right? That's biparentally inherited DNA. You get 3 billion from mom, 3 billion from dad. And so we're diploid. And so we have two of chromosome 1, two of chromosome 2. We're inheriting half from mom and dad. And so the mitochondrial DNA and Y chromosome, those are haploid. Y chromosome is nuclear DNA, but you only get one. You get one Y chromosome, and that's inherited paternally. Your mitochondrial DNA, that's inherited maternally. Okay, only on the mother's side. And the mitochondrial DNA exists outside of the nucleus in an organelle called the mitochondria. That's the cell's power plant. And so if we've been evolving for millions of years, and you've had all these subgroups like Neanderthals, Heidelbergensis, Erectus, you'd expect more diversity in all DNA compartments. And yet, what do we find today? Well, before I show you the answer, notice the slide. The separate ancestry model predicts low genetic diversity. The evolutionary model predicts higher levels of genetic diversity. Brian, can you guess what we find in genetics, brother?
We find
Well, right
lower,
here.
yeah, human genetic diversity is
You got it,
substantially
brother.
lower.
Substantially lower. That's right. And we find one mitochondrial DNA, one Y chromosome. Why don't we find a diversity of them? Right? Especially if the out of Africa model is true, where you have all these different subgroups of hominins accumulating their own differences, and then humans meet up with them, even interbreed with Neanderthals. So there's gene flow there. The mitochondrial DNA and Y chromosome should look a lot different than what they look like if evolution was true. But evolution is not true, and that's why these DNA compartments look exactly like we would expect if creation was true. So notice this. Human genetic diversity is substantially lower than that of many other species. What did I predict, Brian? Animals would have higher levels of genetic diversity overall. Okay, there's always an exception to the rule, but that doesn't overturn the rule. And humans would have lower diversity. Based on an examination of our DNA, any two human beings are 99.9% identical. Isn't that amazing? Any two human beings on the planet were nearly identical genetically. All human beings, 99.9% identical in their genetic makeup. Notice this from the Human Genome Project. We are all remarkably similar. All human beings are 99.9% similar at the DNA level. Evolutionary rescue devices don't work. What's their rescue device? Out of Africa population bottleneck. Oh, well, okay, we're surprised. Humans have low genetic diversity. That's kind of shocking. But what if there was a massive bottleneck event in Africa? Some say 70,000 years ago, others say 100,000, 200,000. Anyways, the point is you have humans, they experience a near extinction event, and so they are reduced to somewhere between 2 and 10,000. Some say 30,000. But Brian, this isn't a one generation bottleneck. This isn't a two generation bottleneck. This occurs for many generations, thousands of years. And what this results in is severe genetic damage. I'll tell you why. If you have a population of between 2 and 10,000 existing in Africa, even broken up into tribes, okay, what you're going to have is severe inbreeding. Inbreeding reveals the hidden reservoir of genetic mistakes that have been accumulating, okay, in a recessive form. The evolutionary model explains the origin of all DNA differences for the most part as being the result of mutations. So that's a lot of mutations that have been accumulating. Us creationists, we look to the majority of DNA differences as being pre-existing. In technical terms, this is called created heterozygosity, that God would have created the original created kinds and humans with pre-existing DNA diversity. And so, no, not all DNA diversity is a result of mutations, but that's the assumption of the evolutionary model. Anyways, so if you have this population in Africa, you have this multi-generational bottleneck. It's occurring for thousands of years. You're going to have all these deleterious mutations coming to the forefront. You're going to have severe inbreeding. You're going to have accelerated genetic degeneration. And we're supposed to believe that this group of highly inbred, genetically compromised human beings suddenly spread out into all parts of the globe and seize dominion over the planet and eventually invent cars and agriculture and these computers that we're doing these live streams. Oh, I'm in Canada, you're in America. I mean, this is amazing that we can do this. This is extremely sophisticated technology. But yet, all of this came from what? Some near extinction event with human beings that would have been genetically compromised? No. And here's the thing, the other objection would be, well, you guys in the creation model, you guys have a inbreeding problem too. Do we though? No, we don't. Adam and Eve are created free from mutations. They're front loaded with genetic diversity. They're created in a state of DNA diversity. But here's the thing, all of the bottlenecks associated with the creation model, they're one generation followed up by rapid and exponential population growth. Okay. Creation, two people, Adam and Eve. Adam and Eve right away start having kids. Their kids start having kids. They have no mutations. They're created with design diversity. There's not an inbreeding problem because, again, I said earlier, inbreeding reveals the hidden reservoir of genetic mistakes. If there's no genetic mistakes, then there's no deleterious mutations to come to the forefront to lead to genetic degeneration. Plus, you have a rapidly expanding population. Then you have a bottleneck 2,000 years later at the flood. You're left with 8 people. Noah, his wife, their 3 sons, Shem, Ham, and Japheth, and their 3 wives. Okay, and it actually turns out that all mitochondrial DNA worldwide can be traced back to 3 major haplogroups, L, M, and N. That's amazing because the Bible says that the earth is overspread by 3 reproducing couples, Shem, Ham, Japheth, and their 3 wives. More confirmation of the biblical account. Anyways, that bottlenecks one generation followed up by rapid and exponential population growth. Then you have the Tower of Babel. Okay, and then eventually the languages are confused and people spread out. So no inbreeding problem for us, but for the evolutionary model, major inbreeding problem. So here's the mitochondrial, right here's the real Eve, the mother of all living. But here is a representation of our genome. Okay, all the chromosomes you'll see at the bottom there, you're either XY, that makes you a man, or XX, makes you a woman. The Y, there's no chromosomal counterpart, so there's no recombination. There is some gene conversion, okay, at the tips, it seems. And the Y chromosome is uniparentally inherited, so is the mitochondrial DNA. But again, as I was saying earlier, Brian, the mitochondrial DNA exists outside of the nuclear DNA. So it exists outside of the nucleus, and that DNA controls the conversion of sugar. So it's involved in very important functions for the cell. The DNA controls the conversion of sugars into energy. It creates ATP. And in each cell, you have many little mitochondria that are working to produce energy for us to what? To live, to breathe, to function. And this energy produced actually sustains our lives and also our bodily function. And so this is important because mitochondrial DNA and Y chromosome, we can look at to help us determine and answer the question of ancestry because they're less messy, no recombination. And so mutations accumulate and we can take the branches formed due to mutation accumulation and we can trace them back to common ancestors, specifically 2, one male, one female. We can take mitochondrial DNA in the world, Brian, this has been done. Look at them and compare them, okay? And build a family tree that points us right back to a single female ancestor. And Eve is largely known, okay? We have a close approximation to her sequence. The mitochondrial DNA is about 16,500 letters, and you can line them up, and you'll notice that 85% of the letters, everyone in the world has the exact same letters. So what does that mean? Well, there's very low variation in the mitochondrial DNA. And so this data matches biblical expectations. And to make it understandable, what was done in this study, Dr. Rob Carter did this study, is it would be akin to if I wrote a book, a manuscript, and I handed it to 100 different people and I said, copy out this book. So you have 100 different people copying out this book by hand, okay? Well, naturally, there's going to be typographical errors in each book. But those errors, spelling mistakes, they're going to be unique to the book, depending on who's writing it. So I take all the books in, and I'm just trying to take the copies and figure out what the original book looked like. Okay. Well, all I got to do is look for the rare deviants, look for the spelling mistakes, the unique typographical errors, remove those from the equation, and then I'm left with a pretty close approximation to what Eve's sequence would have looked like. Brian, does that make sense, brother?
Yes.
Because that's, yeah, okay. And that's what you do. That's what has been done to get an Eve consensus sequence. Okay. So for example, let's say you look at a letter and everybody has that letter. Okay, let's say everyone in the world has an A, right? You got A, T, C, G, and one person has a T. Well, if everyone in the world has an A, but one person has a T, what do you think is the mutation? Well, the T. And the A would be the original letter. And so that's why I say almost all of Eve is known. Now, what we have here is from the paper. This gets a little technical, but it's important. Okay, these are just counts of sequences. Most people differ from each other by about 22 mutations. That's the average. Max is about 120, which is still quite low. And we have good explanations for why it's a bit higher than the mean. And so if we compare everybody on the planet to Eve, most people are within just a few differences from Eve, with African groups being more divergent and then Neanderthals, fossil sequences, being even more divergent. Okay. In a small fraction of the letters, you see what? You see variation. And most of the genome, though, 85% is basically identical. But if the genome were old, you'd actually expect the opposite to be true. You'd expect many more mutated sequences in the genome in general, the Y chromosome, autosomal nuclear DNA. And yet we don't see the kind of frequency in terms of mutations that would be expected if the evolutionary model is true. Now, there are some objections where some are saying that there, and it seems to be largely true, that there is stronger purifying selection in the mitochondrial genome than in your biparentally inherited DNA. Because again, the mitochondrial DNA is extremely functional. Even the evolutionists acknowledge that. The mitochondrial DNA is involved in cell respiration, controlling the conversion of sugars into energy. And therefore, if there's a mutation, possibly even a lower impact mutation, where normally in the biparentally inherited DNA selection wouldn't see, in the mitochondrial DNA, selection might have an easier time seeing it, removing it from the equation. Okay. But nonetheless, this is not an issue for the per generation rate and the long-term substitution rate, because, and I've written an entire technical article with Chris Roop on this, and Dr. John Sanford peer reviewed it for us. But just in a nutshell, the mutation rate, okay, per generation mutations that are coming in versus the accumulation rate or fixation rate. Well, Fatuma, Basic Population Genetics, says this. Notice this quote from Douglas Fatuma in a popular introductory level textbook. The same number of neutral mutations should be fixed every generation. The rate of fixation mutations is theoretically constant and equals the neutral mutation rate. So you've probably seen, Brian, from people like Dr. Zach Hancock, Dr. Dan Stern-Cardinal, they'll say, well, your observed mutation rate is fast, but most of those mutations are weeded out. And so over a long period of time, your substitution rate, it's different than the per generation rate. The fixation rate's a lot slower, and it's too slow for young Earth creation to account for because the timeline goes back further than just 6,000 years. But again, they're contradicting what Fatuma says. This is their own community here. These are their own scientists. That the neutral mutation rate, which I would argue nearly neutral mutation rate, equals the fixation rate. Okay, sure. There's more selection in the mitochondrial genome than was predicted by both camps, evolutionary and creation model. But here's why things balance out for the most part. This is why the per generation rate approximates the fixation rate, maybe not exact, but close to, which puts us at about 6,000 years. Okay. Amazing stuff here. Is because we have a short period of time, Brian, after the flood, where fixation is very fast. Fixation means just to get it stuck in place. You get a new mutation, new allele, that has to spread to every single person in that population. That's species wide change rather than just a single individual passing on some mutations. This is species wide change. So fixation basically today is zero because we got 7 to 8 billion people on the planet. People are spread out all over the globe. If some random guy, let's say some random farmer somewhere in the world has a mutation, passes it on, it's very unlikely that that mutation is going to spread to every single person on the planet. Be very difficult. But in the past, we had a time from creation to the Tower of Babel where fixation was incredibly fast. You have 3 reproducing couples. They start having kids. Humans don't take God's command seriously to be fruitful and multiply and spread out. So they isolate, they don't spread out, right? You get the Tower of Babel, this small scale example basically of one world system where people aren't spreading out into nations. Anyway, so you have at least, a few hundred years there where the mutation rate is fast. The negative conditions associated with the flood would make mutation rates faster for a time, I would argue. And you have in small isolated populations, and even if they're expanding but not spreading out, you have mutations coming about and becoming stuck in place. Rapid fixation. So yeah, the rate is slow today because of human demographics. And the mitochondrial genome, because of its functional nature, has more selection. But again, we have this period of time in our history, according to the creation model, where fixation was so fast that things balance out. And the evolutionary community can't even argue against this because guess what, Brian? They have the same thing. If you have a pen, let's say a pen, evolutionary history is just the tip of this pen, okay? In terms of what we're seeing here with agriculture and people spread out and you have communities and countries and so on. For most of evolutionary history, you have humans existing in Africa in small isolated populations. You have population surges, population crashes, right? These bottlenecks, extended bottlenecks. It's not until recently, according to their model, that people actually expand into all parts of the globe. So they too have an extended period of time where you have fixation being faster. Mutations coming about and becoming fixed a lot easier. It's only until recently where we have a population in the billions and spread out into all parts of the planet. Okay, so yes, there's a balancing out effect. That's why I don't believe that people like Dr. Dan Stern Cardinal, he just put out a video responding to Rob Carter. And I watched his whole video and I considered it. And again, I argue there's a balancing out effect because both models have that period of time where fixation is fast. And so I don't believe their argument works. Okay. That was a bit technical, but it's important because it's what the evolutionists are arguing. Okay. So with all that out of the way, let's move on to our main piece here today. And we are going to run through this. And I'm excited. But before we do, Brian, my brother, did you have any questions based on anything we just discussed there for the last 30 or so minutes?
I've written some things down, but I'll get to those after you get through your presentation.
Okay, perfect. Okay, so we have the human Y chromosome here, another uniparentally inherited DNA compartment. So you'll notice that on the screen here, this is a visual of one of the many papers related to the Y chromosome dissimilarity between humans and the great apes. You'll notice that they have humans as a great ape because they believe humans are great apes, and great apes like chimpanzees, bonobos, gorillas, orangutans are non-human great apes. Okay. We would argue that humans are separate from your great apes. We know that from the scriptures. We saw that earlier in the presentation. So notice here, this data strongly confirms exactly what we went over earlier in Genesis. Okay. So here's your human Y chromosome, chimpanzee, bonobo. This is why I like to say the differences, Brian, make all the difference. Because, yes, we have a lot of similarities in genetics, physiology, anatomy, morphology. We can explain those easily. Creationists explain them as being the result of common design. But it's the differences that make all the difference, because creationists expect some differences if separate ancestry is true. But the evolutionary model, they might be able to account for some differences. But when the differences are too massive and too incredible and too drastic, that's when we see a data point that works towards precluding evolution, meaning making it impossible because they can't explain it. So when you consider overall gene content, size differences, and architecture, because evolutionists want to assume that humans and chimps are related. And so when they see massive differences, they'll just say, well, that's there because of a large deletion or a duplication or a rearrangement. Okay. Because they're assuming all differences are the result of mutations over time. That's you using evolution to prove evolution. So we're just going to consider everything. And when you consider everything without assuming evolution, you have a Y chromosome in humans that is less than 30% the same. Here, as of this year, that this was put out, this paper, it was about 35%. Now, which we're going to see soon, it's less than 30, which is, it's not getting any better for the evolutionists. Anyways, you'll notice the chimpanzee Y is half the size. The entirety of the heterochromatic arm is missing in the chimpanzee. You can see the color code here. So this is heterochromatin. That is a type of DNA that's densely packed. It primarily comprises repetitive sequences like satellite DNA. Okay, it's functional, it's beneficial to the organism. But you'll notice with chimpanzees, it is basically missing. And so the question to the evolutionist is, where did it go? How did it go missing? How was it lost? Okay. Gorillas and humans, when you consider, again, overall gene content, size differences, and architecture, the gorilla and human Y are more similar to each other than either are to the chimpanzee Y. And what that does is it breaks the expected nested hierarchical pattern in the evolutionary model. And so what we actually see here in this visual is exactly what we would expect from the separate ancestry model. We have humans as their own created kind. We have chimpanzees and bonobos going back to their own created kind, or specifically archetype 4,500 years ago. We have gorillas as their own group, and then the orangutans. This is exactly what we would expect. This is amazing stuff. I want to go over some excerpts here from the paper in 2020, Dynamic Evolution of Great Ape Y Chromosomes. Despite a recent divergence of these species, so they're assuming there was a divergence, right? Their Y chromosomes differ just a little bit. No, differ enormously in size and gene content. But they won't even question the evolutionary narrative, will they? They'll just say, well, okay, there are enormous differences. Now we got to go and we need more research grant money to figure out why, if evolution is true, because they're assuming evolution is, it has to be true, because what's the alternative? For example, the chimpanzee Y is only half the size of the human Y. That's why I pointed out earlier. And the percentage of gene families shared by these two chromosomes that split 6 million years ago is similar to that. Notice this, Brian, shared by human and chicken autosomes that split 310 million years ago. That's how big of a shock and an unexpected surprise this was. Puzzlingly, so it is a puzzle to them, in terms of shared genes and overall architecture, the human Y is more similar to the gorilla Y than to the chimpanzee Y. So there's a break in the nested hierarchy. So yes, overall in the biological world, there's a natural nested hierarchy where humans are more similar to the great apes than they are to monkeys and dogs and fish and bacteria. Okay, but when you actually look into the details, there's a lot of discrepancies which tell us powerfully that these hierarchical patterns are there due to design and not descent. Because it's the discrepancies, it's the differences that the evolutionists really struggle with. So here is in 2023, 2024, okay, within that span of time, evolution was just demolished. So they came out with even more sophisticated sequencing data, Brian, on the Y chromosomes. And amazingly, the differences are even more. Notice if you take humans and then go up to your chimpanzee, in this category here, it's less than 30%. Less than 30% the same. That's 26.2%. Okay, and this is why it's so important to focus on the discriminating data, because creationists, we can explain both homology and also significant differences, right? But the evolutionary community, they in a way can account for similarities as being the result of descent with modification, but again, they really struggle with these differences, especially the massive differences. I mean, I was pointing out earlier that even in similar sequences, there's differences in the way they're regulated, the way they're expressed, the way they're turned on, turned off, used. And there's epigenetic differences, differences in alternative splicing. The differences are truly incredible. And so this is a major point against the evolutionary model. Extremely different, the Y chromosomes. And you'd think that the Y chromosomes would be a little more stable due to its lack of recombination. It's less messy. It's not biparentally inherited DNA. It's not autosomal DNA. You'd expect it to be a little more stable, and yet it's the most different chromosome. Notice these incredible differences that I've laid out for people. The difference between human and chimpanzee Y chromosomes are said to be due to a combination. So this refutes a point by Dr. Stefan Frella, who I've been going back and forth with. He's a plant biologist. I actually debated him about 4 or 5 years ago on Modern Day Debate when I first started this. And he's had a written debate in the AIG journal with Dr. Nathaniel Jeanson. So he's been around and he's familiar with the origins debate. And so he and I, we've been going back and forth on this issue. I published an article responding to him on the Y chromosomes. His response is basically storytelling. Okay. He also asserts that it's all gene loss. It's all gene loss. If you actually dig into the literature, okay, they say it's a combination of gene loss, gene gain, and I'll show you why in a second, and large scale chromosomal rearrangements. Remember, these are all assertions because they're assuming that humans and chimpanzees split from a common ancestor. More than 30%, notice this, more than 30% of the chimpanzee Y chromosome fails to align to the human Y chromosome. So if you line the two Y chromosomes up, there's more than 30% of the chimpanzee Y chromosome that doesn't even have an alignable counterpart in the human Y. The portions of the Y chromosomes in humans and chimps are said to have been rearranged, or possibly they're not even related. That question, Brian, doesn't even cross their brains. The chimpanzee Y chromosome, notice this. The chimpanzee Y chromosome has twice as many large palindrome sequences. What's a palindrome sequence? Well, they serve to protect important genes in male fertility. So the chimpanzee has at least twice as many, double the amount as the human Y. Okay. So this is why some in the evolutionary community would say, well, there's gene gain and gene loss. Because after the split, and Stefan Frello, he takes the easy way out. But again, this is just story time. This is like a recipe. Okay, we gotta put together the perfect concoction. Just make stuff up to try and explain how we got from a common ancestor between humans and chimps to humans and chimps. Okay, so from my understanding, what he's saying is, well, maybe they had equal amount of palindrome sequences, but in the human line, they lost half of them. So that's why chimpanzees have double. And then in chimpanzees, all their massive differences are due to loss. But none of this is empirical. It's just trying to come up through storytelling, through guesses. Okay, well, over here, there was some loss in the human line. And then over here, there was a lot of loss in the chimpanzee line. They lost the whole heterochromatic arm. They lost, these genes, this gene family. It's like, well, maybe in the chimpanzee line, they actually added the palindrome sequences. How can you prove either? I mean, these guys like Dr. Stefan Frello, they're just asserting that, well, in the human line, in that instance, it was lost. But wait a minute, they're largely looking to loss in the chimpanzee line due to sperm competition and mating habits. But now when it's inconvenient for them, now they're just going to say, well, in the human line, these sequences were lost because they think it's easier to explain. But again, it's all storytelling. They can believe in this fancy story. They can believe in this rescue device, but it's not science. It's evolutionary philosophy. They're the ones that want to go about putting this forth as if it's science, empirical science, when it's not. Okay, according to evolutionary scientists, mating habits in chimpanzees are one of the major factors in accounting for the massive differences in Y chromosomes. That might help to explain some of the differences in bonobos and chimps. Okay, because with bonobos and chimps, you are mostly looking at gene loss. Okay, and we in the separate ancestry camp, we would say that chimps and bonobos do go back to a common archetype, and we're justified in that. I believe, I think it's a scientific explanation since they can produce fertile offspring, they can interbreed. So at first approximation, we can say, okay, that might mean they're related. Now we can go about explaining the differences. Humans and chimps can't interbreed. Humans and bonobos can't interbreed. Okay. Here's chimpanzees and bonobos. So here's my response to Gutsick Gibbon and Dr. Stefan Frello that say, well, humans, chimps, and bonobos, they can be related. We can account for the differences because guess what? You have to account for the chimpanzee and bonobo differences in a short amount of time since 4,500 years. So why can't you just extrapolate that to include humans? Firstly, chimpanzees are missing, as we demonstrated earlier, the entirety of their heterochromatic arm on the Y chromosome. On their own, these make for remarkable differences between human and chimpanzee Y chromosomes. We as creationists, Brian, we don't have to account for these kinds of differences for chimpanzees and bonobos. They go back to an archetype 4,500 years ago, coming off of the ark, and then they diverge. Okay, speciate, vary. Speciation can happen easily, especially in the design diversity model. All you need is breakaway populations, right? Daughter populations from parent populations, isolation, inbreeding, shifts in heterozygosity to homozygosity, more allelic variability to less allelic variability. And especially in the right kind of environmental conditions, you can have a lot of gene loss. And that's what we see with chimpanzees and bonobos. But they're still way closer than humans and chimps are. Okay, but we don't have to explain anything as drastic as bonobos or chimpanzees losing the entirety of their heterochromatic arm. They have to when they bring in humans. On their own, these make for remarkable differences between human and chimpanzee Y chromosomes. The question is how and when did these colossal discrepancies take place? They're just going to work backwards and they're going to make a story, right? And they got a lot of scientists to come up with some fancy stories. Well, over here, this was lost, and over here, this was lost. And then eventually you get to today. Okay. No, it's a lot more complicated than that. Secondly, chimpanzee and bonobo differences are not sequence specificity. This is an important point, Brian. But multiplication and deletions of large pieces of DNA. These are the types of mutations that we see in real time. They're not far-fetched. Since the differences are predominantly gene loss, which equates to deleterious changes, but human and chimpanzee differences consist of both gene loss and gene gain if you consider the palindromic sequences. Or you can just go with more storytelling like Dr. Stefan Frello does, which it seems. And well, those palindromes were lost in the human line. They want their cake and eat it too, right? It's largely due to mating habits and sperm competition in the chimpanzee lineage and populations. And so there's been a lot of loss there. But hey, there's a little loss over here in the human line too. Explaining differences between the former is explainable. Chimpanzees and bonobos, we can explain empirically. It doesn't take any far-fetched mental inventions to explain differences between chimps and bonobos. While accounting for differences between the latter, humans and chimps, is not achievable. It requires storytelling. And again, they can believe their story. They have that right. But that does not make it science. Protectors of evolutionism can surely believe that humans and chimps are related. But awkwardly for them, this conviction is not scientific. You can see this in my article responding to Dr. Stefan Frello. It is a pseudo-scientific belief, since it is contrary to the empirical scientific data. Defenders of ape-to-man evolution are heavily invested in their evolutionary philosophy. That's exactly what it is. This is why so many followers of this falsified theory have refused to tap out. And that's what we've seen. But this goes back earlier, and this is why it's so important to set the foundation. The Bible predicts, okay? The Bible predicts that in the latter days, there's going to be scoffers. Another consideration here, before we start to wind down on this issue. Remember, it's the evolutionist that assumes all DNA differences are the result of mutations over time. So they see the human genome, the chimpanzee genome, they line them up. Okay, there's a DNA unit, let's say, missing in the chimpanzee line, but it's present in the human line. Well, they're going to conclude, rather than being a created difference, representing the fact that they're not related, they're going to say, well, maybe there's a deletion over here. Okay, and with other differences, maybe there's a duplication here or a rearrangement here. Okay, so they're just storytelling to try and get those sequences back to a starting point where they are basically the same. Here's another point. Proposes a potential genome. So they have to propose an imaginary genome. They got to make up a genome because we don't have. Ask the evolutionist. Show me the fossil of our common ancestor between humans and chimps 6 million years ago. Show me their genome. What did the genome look like? They don't know. It's a guess. They have no idea. They can't even point to a fossil. So they have to propose a potential genome. And then from there, storytell their way to the Y chromosomes that we see today. And they think this is science. No, this data point, even though they've all tried, Gutsa Gibbon has tried several times. She's put out videos. I've debated her on it for 2 hours. Okay. No, this line of evidence really does preclude evolutionism. Okay. And it's all circular reasoning. Assume evolution to argue for evolution. Phylogenetic analyses assumes all DNA differences are the result of mutations. When you see these phylogenetic analyses and they point to a nested hierarchy between various genetic markers and genetic sequences, they're assuming that those markers are the result of mutations. Okay, perhaps they're the result of design. Perhaps this is a reflection of design diversity, and therefore those nested hierarchical patterns are actually there due to design and not descent. Evolutionary scientists can't prove that claim substitutions, deletions, duplications, and you've pointed this out brilliantly as well, Brian, in one of your videos, and other so-called mutations really are mutations. They can't prove that. That's an assertion that they make. The DNA differences that evolutionary scientists look to as mutations can simply be created differences. Without the hypothetical common ancestors genome, right? They don't have it. They're guessing. There is no way to know for certain that these DNA differences are all the result of mutations. And we can show just through DNA function. We've done an entire program, Brian, on junk DNA. Okay, they want to say, well, the majority of this activity, right? Because we know that the genome is active to about 80%, 60% of it is transcribed, right? You got DNA to RNA to protein, DNA transcribed to RNA, translated to protein. There's a lot of activity and a lot of transcription. They want to say a lot of this is spurious activity. It's just noise. One easy, quick rebuttal, okay, to give you as we move on for the audience, just to kind of have that ammo, is if all of this activity really were just noise, well, that would be very wasteful of energy and resources for the cell. Because, firstly, transcription is a very energy intensive and resource intensive process. Every time you add a single nucleotide to a growing RNA chain, you're consuming what? Energy. You're consuming ATP. So if this really was mostly just noise and spurious, not really beneficial functions, then if evolution were true, evolutionary mechanisms like natural selection should have weeded out all of this trash and garbage over time, because that would be smart, wouldn't it? If all of this noise is wasteful of energy and resources for the cell, then selection should have done something about it, should have got rid of it. Okay. Just like if our dressers are getting too full of clothes where you can't even shut the dresser anymore, the drawer, because socks and shirts are, hanging out of it, you're going to go in and clean up the mess because it's messy. Clutter is not good. So you're telling me that selection and mutations and other evolutionary mechanisms can take a single celled like ancestor into a whale and human over time, but they're incapable of weeding out all of this garbage? It's just more evolutionary philosophy. And so let's stop there. I do have a whole section expanding on the LM and N haplogroups. I'm not sure if you want me to get into that or kind of save that for the Q&A period, Brian, but that wraps up my portion on the Y chromosome, brother. I can expand on anything said or go through more slides. I give a presentation. I have one on my channel titled The Scientific Evidence for Adam and Eve, and I go for about an hour and a half. So there's a lot of data here. Guys, I'm telling you, it is a great time to be a biblical creationist. There's a lot here. This is just a sneak preview, really, into all the amazing data that confirms separate ancestry.
Awesome. And actually, those, I guess while you already got the screen up, well, yeah, we'll keep it sharing so that way you can set to show what you need to show on that. But the three haplogroups, do you want to explain that a little bit more? Me and Matt talked about it briefly, and we did look at the image and stuff too as well. But do you want to kind of like talk about that a little bit?
Sure, it'd be my privilege, brother. So right here is a phylogenetic tree of mitochondrial DNA. Dr. Nathaniel Jensen, he built this tree. Okay, you take the data, represent it through a phylogenetic tree building program, and this is what you get. And so this is real data. These branches, what do they represent? They represent DNA differences or mutations. Okay, so these are mutations over time in people groups, all kinds of people groups here. You'll notice all branches, so all DNA diversity worldwide, go back to three major haplogroups. These aren't even contested. The evolutionary community will acknowledge these groups. L, M, and N. Well, think about the creation model that we explained earlier. You start with Adam and Eve. They have kids. Their kids have kids. Then you have a bottleneck 2,000 years later, which is 4,500 years ago since the flood. And the earth is overspread by three reproducing couples, Shem, Ham, Japheth, and their three wives. But remember, mitochondrial DNA is only inherited on what? The mother's side. It's maternally inherited. It's uniparentally inherited DNA. Okay. And so we would be going back technically to Noah's three daughters-in-law. And that's exactly what we see here. This is amazing confirmation. I mean, this is biblical history. We've confirmed biblical history. It doesn't get any better than this. Of course, again, Second Peter 3, scoffers are going to come about and they're going to try and fight the data because the data points to the Bible as being true. So here's the three arrows, three major haplogroups, and all DNA diversity and mitochondrial DNA goes back to these three major starting points, which could certainly be Noah's three daughters-in-law. Now, there are objections that I can go over, and I'm happy to do so, leaving no stone unturned. But before I do, did you have any questions on the tree, brother?
Not really. About the three haplogroups go back to the or it matches very well with the Bible in terms of the three wives of Noah's sons. Is that correct?
That's right. Yeah. It fit.
Awesome.
See, here's the thing: the data did not have to look like this, right? These are three major groups. Even the evolutionary community have acknowledged this reality, and all DNA diversity worldwide, all these branches, which represent people groups and mutations, they all go back to three main women. Okay. Here's the objections. Oh, well, this is an unrooted tree. Dr. Nathaniel Jensen, he didn't root the tree. Well, what the critics call an unrooted tree actually has a root. And you can ask Dr. Jensen himself about this. I've asked him several times just to confirm. I want to make sure that I'm representing people correctly. And he would say this is the Eve position, right? The whole tree basically represents Eve, and Eve would be somewhere in the center, right? You can pick one of these points. He picks a specific one because again, if these represent Noah's three daughters-in-law, those DNA differences would be traced back to, in the pre-flood world, eventually Eve. So that their sequence could approximate Eve's. When guardians of evolution say unrooted, what they mean is you have to follow our evolutionary assumptions, right? They make the rules, Brian. Apologists of evolution assert that we have to have an evolutionary outgroup. This is a form of circular reasoning. Remember, evolutionists assume that Neanderthals could not have gone back to Adam and Eve. They assume humans and chimpanzees are related. They assume that Africans are older. There's a lot of assumptions there. In the three node model, M and N. So they'll say, this is their main objection, Brian. They'll say, well, L, M, and N could not have been contemporaries. They could not have existed at the same time because M and N had to come from L because of the differences. Okay. And when they root it based on their evolutionary assumptions. But here's the thing. They could have lived simultaneously. This is a weak objection. It's a straw man, essentially. Since the lifespans in Genesis 5 are very long and overlapping, it's plausible that L, M, and N were contemporaries. And if you take one of those charts, a lot of your creationist organizations, and I'd like to get a professionally done one, they have these charts where they show all the names of your pre-flood patriarchs, right? Adam right up to Noah, and then after the flood. And you can see the overlap. You can see just how many people after so many generations existed with Adam, their great, great, great grandpa. I mean, that's amazing. If you're living 900 years and your kids are having kids and your kids' kids are having kids, there's a lot of overlap, isn't there, Brian? Overlap that we wouldn't have today. If our great grandfather only lived to, 85, 90, well, there's not really going to be a lot of overlap. He's not going to exist at the same time as my kids or my kids' kids. But if Adam is living 900, for example, I mean, like your kids could be hearing stories from Adam. Isn't that right? That's right. So, and you can see that really well in a visual if you see all of the ages lined up when they were born, when they died. Okay, so yes, L, M, and N could have been contemporaries. Genesis describes long lifespans in the pre-flood world. Therefore, we should expect overlap in people during this unique time. They're basically making this argument assuming that the Bible's account of pre-flood ages is false. So it's a straw man. They're not steel manning our position. Therefore, we should expect overlap in people during this unique time. This means Noah's three daughters in law could have each come from different families, had different mutations, and could have all been different ages. This means L, M, and N could have lived at the same time. To say otherwise is to misunderstand the biblical model of ancestry. I put out recently an article responding to all these objections and also a couple videos because I've been going back and forth with Dr. Stefan Frello on this. So here's the assertion. But Dr. Jensen has no root on this tree. This is wrong. Dr. Nathaniel Jensen does root the tree. He roots it on the L node as Eve / one of Noah's three daughters-in-law. Again, one of Noah's three daughters-in-law could have a mitochondrial genome that closely approximate Eve's because all this DNA ultimately goes back to Eve's anyways. So I should make this clear. Notice these short branches connecting L, M, and N. Those are short. That represents fewer mutations. Okay, Brian? Notice the branches radiating out. If you can see my mouse here. Notice the branches radiating out. They're longer. So what does that represent? More mutations, more time. Okay. What does that suggest? What can I predict? Well, the shorter branches connecting L, M, and N would represent pre-flood history because those are mutations that Noah's three daughters-in-law have, and those are mutations that would have been accumulated in the pre-flood world, right? Longer lifespans, fewer generations, fewer time. This makes sense. This is beautiful. And then you have more time, more generations, shorter lifespans post-flood. And you see what? More mutations, more time, longer branches. Okay. And so again, those shorter lines, L, M, and N being separated by shorter branches suggests that, yeah, they could have lived at the same time and they could approximate Eve's sequence. This is because one of these wives of Noah's sons could have mtDNA that is very close to Eve. The most important fact is that testable predictions have resulted from the placement of this root. And so that basically resulted in Dr. Jensen saying, maybe we can read the history of civilization off of this tree. Okay, mitochondrial DNA represents a uniparentally inherited DNA compartment. Wait a minute. Isn't there another uniparentally inherited DNA compartment? That's right, the Y chromosome. Okay, hence why Dr. Jensen wrote a book that is behind me called Traced, which is basically one big giant fulfilled prediction of this. Here's the other major objection. One of the three nodes has to be the great. Can you hear me, brother?
Yeah, I was fixing to say something real quick. I don't have Nathaniel Jensen's trace book, but I do have his replacing Darwin here. And this, I think this book is the book that has his diagram in it too, isn't it? The one that you show.
That's right. Yeah. He makes those predictions in that book.
Right. And Trace basically shows that these predictions have been fulfilled. I need to get that book, his book on Trace.
Yeah, and Trace differs in the sense that it deals a lot with population history and, you know, which populations came from which, and it basically just traces back the history of civilization in a way that demonstrates it all fits within about 4,500 years since the flood.
Right.
And so they'll argue, well,
oh, go ahead.
Yeah, go ahead, brother.
Well, I was going to ask a question before you moved on to the critics' objection here. What does L, M, and N stand for exactly?
They stand for basically your three major haplogroups that you have mutations from people worldwide, right? As people spread out, they add new mutations and new alleles and basically more haplogroups. So you get an explosion of new haplogroups over time. But the three major haplogroups are your largest groupings that people can fall into. And that's why you see all those DNA differences that have accrued over time from different so you get major haplogroups and then you get haplogroups and then more haplogroups. You can see that here, like over Down here would be, we would argue, here's a haplogroup, but that represents a post-Babel haplogroup. Okay, so unique mutations, unique differences that make up certain haplogroups. Like the HVR haplogroup, that would be your most common in Europe. And so you can look at that and you can see genetic markers that are unique to that specific group.
Are um uh are this associated with bottleneck? Is that what you get with it? Like you can trace this all the way back to these three particular haplogroups. Is that what you're saying, that this is a bottleneck?
Yeah, in a sense, because that would be the starting points. So basically, these three groups here, these three nodes, those would be the starting points of all other haplogroups, all other people groups. So if that makes sense, right? So these are nodes. Think of it as like a family tree. Here's the starting points. Noah's three daughters-in-law. And then all other groups, all other people stem from them. And this is where you get other haplogroups too. So sub haplogroups, HVR haplogroups, um, L would be like your African line. A lot of your African DNA differences would fall in L.
Could
M
you
and N would
explain
comprise different groups, Europe, Asia. Yep. Brian, go ahead, brother.
Well, I was gonna say, could you explain to everybody I know what a bottleneck is, but could you explain to everybody what a bottleneck is, so that way everybody kind of can understand what do we mean when we say that there's a bottleneck?
Yeah, bottleneck is basically, so let's say in the out of Africa bottleneck, you have a large population. How large? I don't know, maybe 200,000 people, maybe a million. Okay. And because of a near extinction event, you have, so if you think of a bottle, right? You got the narrow neck and then the fatter part. Okay. So you have a lot of genetic diversity in that population of, let's say, a million strong. But when a near extinction event reduces that population to, say, 10,000, well, now you have a narrowing of the genetic diversity, right? Where you had a lot of heterozygosity prior, because of the bottleneck, you now have more homozygosity. And that's why the evolutionary community, they need these bottlenecks to explain why there's such low genetic diversity in people groups worldwide, because they need that bottleneck to take what? Levels of heterozygosity, which is more DNA diversity, down into more levels of homozygosity, okay, which is less allelic variability. And so that's basically what a bottleneck will do. It'll take a population that has a lot of genetic diversity and it'll reduce it. Then there's
Uh,
founder effects, right? Founder effects is a little different. We have a small group that isolates itself from the main group. And then from that small group, like with Neanderthals, that small group starts having kids and their kids start having kids. And so that group is unique. And so you have founder effects. That group's going to be unique in genetics, morphology than the parent group.
how does evolutionists justify bottlenecks exactly you mentioned they think that there was a bottleneck in Africa, which is where they think everyone came from, right?
Yeah.
So how do they justify that bottleneck exactly? I mean, I know they don't believe in a global flood exactly. So how do they fit that in within their paradigm?
Yeah, good question. They struggle. I mean, they're largely looking at the genetics. And because the genetics are low, because the genetics don't really fit the evolutionary narrative, that demands a bottleneck. But do they see reflection of that bottleneck in the fossil record? Not really. I mean, they'll say mitochondrial Eve, okay, which all women today come from, and Y chromosome Adam, who's technically Noah, all males descend from, they'll say that those two individuals existed in a greater population. But they're just making that assertion. They don't have that evidence. They don't have it in the geological column. They don't have it in the rock record. They're basically looking at the genetics and they're working their way backwards in a way that fits the evolutionary narrative, right? For example, that DNA barcoding study that came out, which Matt, he's an expert at, that's been a lot of his research.
Oh, I didn't
It's
talk to
funny.
I didn't talk to Matt about that. I should have because that was something when I first heard about that. I thought that was pretty interesting. I don't think I actually talked to Matt about that.
Well, in a nutshell, the Rockefeller study, they determined based on genetic diversity in these highly conserved mitochondrial genes, like the CO1 gene, there's low diversity in animal species worldwide. And so they concluded that all animal species today came into existence at the same time. And they'll just assert through a massive bottleneck. Yeah, that makes sense. We understand there was a massive bottleneck 4,500 years ago where you have arc animals, and then all species today go right back to what? Those original arc animals. That makes sense. All extant species originated, from those original arc animals because those arc animals come off, and then you have speciation events over time. And so they'll say, but this happened 200,000 years ago. But this is circular reasoning. They're not getting that from the genetics because the real time, observable, repeatable evidence from genetics tells us that this happened about 4,500 years ago. But they'll say 200,000 years, largely because of their evolutionary assumptions, right? They look to the geological column and they have beliefs about when certain divergence events took place. They have their evolutionary narrative. And so they have to fit the genetic data with the data from the rock record. And so there's a lot of circular reasoning going on. And so to answer your question, yeah, they don't really have good justification for their bottlenecks. The
Would you say
genetics
that
does point to bottlenecks, but it points to biblical bottlenecks like the flood, Babel, and creation.
Would you say that bottlenecks make more sense in the biblical worldview? Because we have things like the Tower of Babel, we have the global flood with Noah and his three sons and wives, and we also have, of course, creation itself. So, would you say that the bottlenecks make more sense in the biblical worldview than it would in the evolutionary one?
Oh, 100%. Yeah, I mean, that study where it says all animal species are basically the same age based on the CO1 gene, that fits with the flood bottleneck. Human beings have low genetic diversity. That fits with both the flood bottleneck and the creation event of Adam and Eve. Allele frequencies, okay, the fact that you have a lot of genes in common, but then you have unique mutations in people groups worldwide, that goes right to Babel. Because what happens at Babel? Well, everybody is uniform. Then God confuses the languages and then they spread out to different parts of the planet. And then different groups start accumulating their own independent mutations. And that's why you have specific mutations in what? Africa, specific mutations in Europe, in China. Same thing with the Y chromosomes. You have geographically specific Y chromosomes in China, Africa, Europe, Australia. This is all evidence of the post Babel dispersal event. And so, yeah, 100%. The data fits far better with the biblical model, brother. That's a great
One of the
question.
one of the things that I noted, genetics was not an actual study at the time of Charles Darwin, and so now that we have genetics, and the more we study genetics, the more it confirms what the scripture says, and so that always strikes me as interesting is that the genetic data is actually on our side. It looks like, and the more and more we investigate it, because evolutionists, you can't make predictions from genetics as much with evolutionists as you can with creation, because in creation you can be able to make these kinds of predictions and see them validated within genetics. With the only predictions that get confirmed with evolution and genetics is when they, and both creation and genetics says, or both creation and evolutionists say the same thing, like you, what you call agnostic evidence or agnostic uh, arguments, that both sides would say the same thing. And that's the only time you ever see any kind of fulfilled predictions, because what they thought on the Y and X chromosome, they thought, well, the X and Y should start out with the same size, the same percent similarity, and all that stuff. And then they find out that that was not the case.
That's exactly right. And that's why they struggle with all of this data, brother. I mean, what you said there, you nailed it. And that's why this right here on the screen is such fascinating data, because we're able to trace all people right back to these three major mitochondrial DNA haplogroups, right? And that's why you find these groups going back to groups in Africa. From these three major haplogroups, this is where you get, African lineages, Europe, Asia. And so it's all a reflection of true biblical history. And that's why all of history, the history of civilization, can be traced right back to the flood bottleneck.
Did you want to go
And
ahead
what
and
does the Bible
continue?
say? The earth was overspread from those three couples.
Right. Did you want to go ahead and continue with what you were saying previously about the critics? I just had some questions on that before we moved on, because I know that we're going to be pushing forward. So I just wanted to clarify some stuff.
Absolutely, brother. So right here, uh, this is the next one. So this challenge right here, one of the three nodes has to be the great, great, great granddaughter of one of the others. This challenge is based on a straw man. The branch lengths of the mitochondrial DNA phylogenetic tree reflect DNA differences. These DNA differences are the result of mutations. Mutations are transmitted per generation. So they're assuming right off the bat that if there's more mutations, then it has to be this many generations, more generations. Okay. Genesis 5, though, lists the paternal generations between the creation event and the flood. Very few generations passed. Due to the long lifespans, remember I explained earlier how you'd have a lot of overlap, and late childbearing ages of the biblical patriarchs. I mean, some biblical patriarchs could be having kids at 300, 400, 500. That's amazing. Only roughly 10 generations passed in approximately 1700 years. Since men were living long, it's safe to say women were also living long, right? So they'd be having kids later in life too. Both paternal and maternal lineages would be expected to have long lifespans. Therefore, L, M, and N, yes, they could have been contemporaries. And so, no, Noah's sons did not have to be married to the great, 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 great granddaughters of one of the other. And you can really see this if you look at a visual of all your biblical patriarchs and just see how amazing the overlap really is. And so, your final and probably most used objection has to do with fossil sequences. You've probably heard this, Brian. This is a frequently used argument. I've been debating for years now the critics on Neanderthal DNA. I've done several formal debates on this topic. I've studied it extensively. And so notice what happens when you add in, Brian, the fossil sequences. So fossil sequences would be Neanderthals, Denisovans, right? Hominins. The branches go out even further. So the evolutionists say, oh, see, when you bring out, so over here, right? Three nodes, L, M, and N. This is where you point out this is primarily the African branch. Okay. You put the fossil sequences, the branches go even further. So the evolutionist argument is basically, well, that means Neanderthals are older, and there's no way you can fit this tree within 4,500 years. Does that make sense? To steel man their argument, you add in Neanderthals, it takes Eve back further than 4,500 to 6,000 years ago. They say this will go back, 100 to 200 or so thousand years ago, even further, to explain those differences. And so, no, easy explanation. All right, Neanderthals, they were a unique people group. They had more mutations. They were different in their genome. All you need is more mutations accumulating in a short amount of time. And the evidence does suggest, contrary to what the critics say, that Neanderthals were hypermutating. Okay, I've got a whole section in my book, Special Creation, on Neanderthal phylogenetics, but I want to give you a shorter, more concise response. So I want to flip to this slide. Okay, Neanderthals, yes, they were inbred. So we have genetics from late stage Neanderthal where they were the most inbred. They were on the verge of extinction. They were 40% less fit than modern humans. They had these massive runs, Brian, of homozygosity. What does that mean? Well, you have these massive stretches of identical letters, right? This goes back to basic alleles in genetics. If you're capital A, lowercase a, capital B, lowercase b, you're genetically heterozygous. There's differences there. If you're capital A, capital A, capital B, you're what? Genetically homozygous. There's no diversity there. So Neanderthals have these massive stretches of identical letters, very, very inbred. So your critics, like Dr. Dan Stern Carter, knows they can't be hypermutating because they're inbred. That's a contradiction. It's got to be one or the other. Well, Neanderthals could have been hypermutating in early generations, right? Could have been just a small family. And I point this out here. The founders of Neanderthals could have been a single wandering family, which means they likely had a coalesced mitochondrial DNA sublineage. Founder effects, we talked about this earlier, in the case of Neanderthals. So there is a difference between founder effects and bottlenecks. Could have been a small family breaking off from a larger population. Could have contained only a single mitochondrial DNA lineage. Then the founder population, the founder effect population could have even been siblings. It's not necessary, but could have, can't be ruled out. So point is, you have a single wandering family, you could say, or a wandering group that break off even before Babel. And they move into a less than ideal part of the planet. As their history is taking place, they could be expanding in population size, breaking off into different tribes. They could start off in the early part of their history hypermutating, but then eventually they become strictly isolated, different tribes. They're not growing in population, they're inbreeding, and so they become homozygous. Okay, so this is why I say here, new mutations would also rapidly become fixed, since faster mutation rates in small isolated populations have a greater chance at becoming fixed, resulting in even more genetic distinctiveness. So yes, Neanderthals are genetically distinct. They map out differently phylogenetically than modern humans, but they have totally different histories. They existed in different environments. They had different mutational events. But most importantly, this is huge, Brian. And I did a whole presentation responding to Dr. Stefan Frello the other day. And I went through all the resources on Google showing right from their literature that radiation rocks can greatly speed up the mutation rate. I went over right from their literature how cave dwelling creatures, because of the factors associated with living in caves, can increase mutation rates. Okay. I went over all the facts showing that smaller populations, isolated populations, have faster rates of fixation. Anyways, what do we see? We have evidence that Neanderthals existed in isolated populations in caves and in areas where they'd be subject to radiation rocks. What do I mean by that? Well, Brian, you're more than familiar with this, brother. All too familiar, as you've been working diligently on the heat problem. So the critics say the flood, the global flood, would have resulted in what? A lot of heat, a lot of radiation, a lot of energy. So right after the flood, what are you going to have? You're going to have radiation rocks. in specific parts on the planet. And if you have a small tribe living in an area where they're exposed to radiation rocks, that those rocks would have the most radiation because it's right after the flood. The flood would produce heat and radiation. And those groups of people that are living there after generations, their mutation rate is going to be greatly sped up. Okay, so notice this. Living in caves can accelerate mutation accumulation due to several factors. Factors include reduced selection pressure against slightly deleterious mutations, so they're going to flourish, exposure to certain chemicals, and especially a lot of these caves formed rapidly from the flood. And then you have Neanderthals. It's a sad state of affairs, but the effects of the flood, these post-flood conditions, they're exposed to these radiation rocks, and they're accumulating a ton of mutations in just a few generations. Notice this: radiation within the cave environment and altered DNA repair enzymes due to unique cave conditions. And so I have several papers that go over the real-time evidence where real families, okay, this is observable evidence, they, for a number of reasons, radiation, chemotherapy, they were hypermutating. They were passing on like 7 times the amount of normal mutations. And they had errors in DNA repair enzymes. So here's the thing. All you need is Neanderthals being exposed to radiation rocks, living in caves right after the flood. They could have mutations in DNA repair enzymes. If DNA repair enzymes break down due to deleterious mutations, Brian, what that means is you're going to have fewer repair enzymes to repair mutations. Therefore, more mutations are going to accumulate generation after generation. So, what evidence do we have? Radiation rocks. They existed in small, isolated populations, which means faster rates of fixation, which is what? Species wide change. So Neanderthal groups are changing rapidly. Okay. And they were also subject to harsh environmental conditions.
I
Again,
want
you
to.
can see this all throughout the conventional literature. What I'm saying here is true. They admit that radiation rocks and caves and errors in DNA repair enzymes can greatly speed up the mutation rate. Go ahead, Brian.
Well, I was gonna say, I wanted to talk a little bit more about the mutation rates.
Yeah.
Does that got to do with genetic entropy?
Yeah, genetic entropy is the reality that the mutation rate is high and natural selection is limited. And so the rate is high. Most mutations are nearly neutral, which means they're slightly deleterious, but they're invisible to selection. That means they're subject to genetic drift. They spread, they degenerate, and species descend into genetic sickness.
All right. And have you read Dr. Dan Cardinal's paper with Zach Kangok?
I've gone through it. I've also asked Dr. Rob Carter about it. I've sent it to Dr. Sanford, and I also sent it to Paul Price, and he went through it all. And so he's given me a little bit of feedback. So we are working on that paper. Yes.
Okay. So
I
have
have
you
to
got it?
read through it a little bit more comprehensively, though, when I'm ready for a response.
Okay. Okay. Then we'll keep that for later on. I got the
Well,
paper on my computer, but I haven't actually
I
read
could
it.
say this because I yeah, I do have a preliminary response because the paper assumes way too much about beneficial mutations and compensatory mutations. Okay. We understand that beneficial mutations are rare. And even when they do happen, genotypically, they're largely reductive, the breaking of pre existing systems. Okay. And so they're functionally compromising to organisms. So Dan and Zach would have to assume that there are way more non-reductive beneficial mutations than there are in order to compensate for the data. So there are still some assumptions going in to their paper that I don't believe fit reality.
I remember
That's a
in one
preliminary response.
right. I remember in one chat they said nobody's responded to their paper yet. So I was like, well,
Yeah.
I haven't really read it yet. I mean, I've got it on my computer. Dan sent it to me via email, but I haven't actually had a chance to read it. I know Dr. Rob Carter has gone through it a little bit. Um, but I know he gave me a little bit of a feedback on it a while back. I have to search for that email, but yeah. All
So
right.
much work, so little time, right, brother?
Right. But would you say that this mutation rate agrees with the young Earth position and model?
Absolutely. The known mutation rate, especially considering the fact what we covered earlier, that the mutation rate approximates the fixation rate right here. Even with ongoing random loss due to genetic drift, classical population genetic theory states that over evolutionary timescales, a neutral mutation fixation equilibrium point will be reached. When this occurs, the neutral mutation rate will equal the fixation rate. We have this unique period in our history where fixation is so fast after the flood and before the Babel dispersal event. We have isolated, a smaller isolated but expanding population. Okay, which means fixation is going to be fast. And so that balances things out. Even in the mitochondrial chromosome. Yes, there's more purifying selection in the mitochondrial chromosome, Brian. Okay. And fixation's slower today. But because of that unique period in history in the creation model, things are balanced out. Where I would argue that the observed rate, which is fast, the observed rate is what? Okay, you take grandmother, mother, daughter, count up their differences in the mitochondrial DNA, right? And then you get a fast rate. That's the observed pedigree method. And that fits beautifully with the creation model. It's troublesome in the evolutionary model because they need to extend that back a couple 100,000 years. And with Neanderthals, that rate is even faster. It could be 10 times or more faster. And why? And think about it, Brian. You're doing a ton of work on the heat problem. So the critics, they say we have a problem with Neanderthals, but then they go and constantly use the heat problem argument when the heat problem and the radiation problem actually works to answer their Neanderthal challenge. Because Neanderthals were isolated populations that existed in less than ideal conditions. They were exposed to radiation rocks from the flood. They were exposed to caves that had more radiation. And since they existed in this way for many generations, what does that do? Speeds up the mutation rate. So even though the Neanderthals branches are longer, that's not because they go back further. That's not because the Earth is older than 6,000 years. That's because they accumulated more mutations in a short amount of time.
Since mutations can lead to speciation, do you think that this would also be evidence of rapid speciation following the flood?
Great question. Yes. Yep. I do believe. See, here's the thing there's evidence that speciation rates, which evolutionists call evolution in the literature, okay, you can look it up. Does higher levels of heterozygosity mean more speciation? It'll say yes. So what do we believe? At creation and then at the flood, organisms have higher levels of heterozygosity. So animals come off the ark, they have more heterozygosity. They also are existing in harsh environmental conditions. It's widely understood that environment dictates phenotype in many ways. Like Neanderthals were cold adapted. They are adapted to cold ecoglacial environments. So yes, speciation rates would have been rapid after the flood. I would say those 1,000 years after the flood, you'd have all kinds of variations coming about, which is why we can see depictions in things like Egyptian archaeology of essentially modern species like lions and leopards and other felids and because those species for sure came about rapidly after the flood, no doubt, brother.
Awesome. Um, all right. Um, you showed that chart earlier on the Y chromosome. You mentioned like maybe an earlier study that showed that we are more or at least more similar to that of a gorilla than that of a chimpanzee. Do you remember what the and maybe you can bring it up as well? What the paper says on the similarity between humans and gorillas on that paper, as far as the more up to date study.
Yeah. So if I understood your question right, we have the older study, which shows humans here and gorillas are more similar to each other than either are to the chimpanzee, right? So that's a break in
Right.
the hierarchy. And they even admit this in their paper right here. The human Y is more similar to the gorilla Y than the chimpanzee Y. So, they admit that. And then here's the newer study that you're referring to.
Yeah.
And so if you go down here to gorilla and work your way up to human, let me see, human to gorilla. Let's go human to gorilla. 25.1%. Gorilla to human, 23.2%. Chimpanzee to human, here's 45.1%. Human to chimpanzee, 26.2%. I guess there's a range because of how they're comparing it, human to chimp or chimp to human. I mean, remember the human and chimp, the Y chromosomes are half the size, but from what I've seen studies, they're going with that lower number. Do you have any input on that, on those differences in those numbers, brother?
Yeah, I'm thinking that the lower number, at least as far as chimp and human, might actually be accurate because of the like you said, it's half the size of the human, the chimp genome. So the 45% could be what is there being compared with it. It depends on whether you're comparing a chimp to human or human to chimp. At least that's my
Yeah.
thinking on it. I don't know if you have some idea that is right. That it just depends on whether they are comparing human to chimp or chimp to human. Would you say that could explain the range there?
I think so because again, there are size differences and
Right.
there seems to be a range. Like if you go chimpanzee to gorilla here, you have 38.1%. And if you go gorilla to chimp, 19.8%. So the lower end is 19.8. If you go orangutan to orangutan, notice how high they are. 87
Here's
orangutan.
a
This is how you know you're on the right track, right?
right.
Orangutan to the same species, 100%.
Yeah,
Different
so
species, 87.4.
if you look at one of the interesting things that I did find whenever I began studying it, I realized that this matched, I mean, 100% matched the creation model of the separate ancestry that you mentioned earlier on like bonobos and chimps are related to each other, but humans it's its own kind, and the other great apes are their own kind. And I noticed that when I looked at the chart. If you look at the chart, the two that are most similar to each other, the two that are most similar are the two species of orangutan. And then you look at it again and you see the second most similar are the bonobos and chimps. So the
Yeah.
two things that we know are very close related are the orangutans, and the bonobo and chimps are not as related as the orangutans are to each other, but they're closely related. And so they're scoring the next highest, and then all the others score like really low as a result. And it's like, okay, this looks exactly like the separate ancestry model, and these people were not even trying to defend that model at all. In fact, they spent most of the paper trying to justify why there's such a difference here. But
Exactly. It's a lot of storytelling, hypothesizing,
would
untested.
you agree
What you
though
just
that
said, you nailed it.
would you agree that it does actually look like it matches that separate ancestry model?
100%. I mean, right here is our model. at Stand for Truth Ministries that we hold to, because some creationist organizations might differ on how many kinds there are in the great ape family. And so you see orangutans, right? The two major orangutan species, they're their own, chimpanzee and bonobos, gorillas, and then your Australopithecines, an interesting group there, extinct. So you have, based on that data, humans as their own group, orangutans as their own group, bonobos and chimps, which are close, and we can explain. We went over that. Gorillas are unique in many ways. And so you have multiple created kinds here. And so this study, like you said, brother, has actually helped to confirm and validate the separate ancestry model, because your orangutans are the closest. Chimps and bonobos are close, right? Chimp to bonobo, 65.3, bonobo to chimp, 60. But we explain the nature of those differences too.
I was just gonna ask you, what is Erica's response? I know you debated her a little bit on an open mic night one night on this very paper. What was her response? I know that the paper tried to argue for sperm competition, and I just want to see what your thoughts are there. And I think Erica in the open mic debate with you brought up the differences between bonobos and chimps. So, do you want to respond to a few things there?
Yeah, and that's where I went over here in these slides, because I wrote an article which I recommend people check out, demonstrating that differences between chimpanzees and bonobos, they are readily explainable in 4,500 years since the flood, diverging from a common archetype. But the second you bring in humans, it's a lot more troublesome because the differences are much more massive and unrealistic, like the loss of heterochromatin, the complete loss of heterochromatin. Many would have to invoke both gene loss and gene gain, right? And chimpanzee and bonobo differences are not sequence specific large pieces of DNA. We really only have to explain deleterious changes, but not deleterious changes in the sense that, hey, this creature here lost the entirety of its heterochromatic arm. Like, that's sure, that's loss, but that's massive loss. And they got to explain that. And so their challenge is largely philosophy. It's in my opinion, it's an extrapolation fallacy, because, well, you guys have to explain chimpanzee bonobo differences. Okay. So when you do that, why don't you just extrapolate that to explain human differences and Y chromosomes too? Right. So they want to bring in humans with the chimpanzees and bonobos and be able to account for all those differences. We as creationists, we're going to have to account for the chimpanzee and bonobo differences, but for the reasons I've provided. And I recommend people check out this article I wrote. Okay, those differences are readily explainable in an empirical manner. But once you bring in humans, there's more hoops that need to be jumped through. It's more unfeasible in explaining the differences. Where sure they can believe, they can extrapolate, okay, but that's just a belief. That's just their evolutionary philosophy. What we have is the data. We see the massive differences. It fits nicely with the separate ancestry model. Okay, that's the data. They're the ones that have to invoke a lot of fancy storytelling. And we have justification, Brian, because chimpanzees and bonobos, they can interbreed, they can produce fertile offspring. So we can look at that and say, okay, that's a good first approximation in saying they're the same kind. Now let's look at their differences and account for them. But the
No,
evolutionary community, they don't even have that first approximation for humans and chimps, because guess what? Humans and chimps can't interbreed.
when I asked Dr. Tompkins about that, he actually told me that chimps and bonobos rarely they interbreed with each other quite often in captivity when they're held in captivity. The reason why they don't do it in the wild as much is because they are geographically separated from each other. And, there's a big old river in the middle, like Congo River or something like that, between chimps and
Right.
bonobos. But in when they're put in captivity, they actually do interbreed with each other. There's evidence of that. There's even evidence
Wow, it's
of
amazing.
bonobo DNA in chimps. So, so that they know
Wow.
that they can, and because they can interbreed, that definitely means that they are the same kind. Although the fact if some two that don't interbreed does not necessarily mean that they're not part of the same created kind, because there could be other reasons behind why two different animals can't interbreed, like size difference, for example, like in the case of the lion and house cat. There's a forty
Right.
percent difference in size. As
Mechanical limits.
yeah,
Mechanical limits.
yeah, that and the fact that the house cat is not exactly in the same level of the food chain as the lion. So, but there could be other reasons for it. But if they do interbreed, that definitely means they're part of the same credit line, beyond a shadow of a doubt, because
Right.
that is what the Bible tells us.
Well, that's an excellent point you make. That's why we, as biblical creationists, we have that first approximation. They can interbreed. Now, let's go into our work and let's explain the differences in Y chromosomes between chimpanzees and bonobos. Okay, because we have that first approximation to go by. The evolutionists, they don't even have that first approximation to say, well, look at humans and chimpanzees when they're in captivity or they're held together, they interbreed and they have fertile offspring. No, they don't have that. And in addition to that, they have the problem of massive differences. Chimpanzees have double the amount of palindrome sequences. Chimpanzees have lost the entirety of their heterochromatic arm. 30% doesn't even align between the two, half the size. It's a totally different ball game. I would argue, Brian, and I wonder if you would agree, it's apples and oranges. Bonobos and chimps, let's have some fun with that. Okay. And we got the first approximation. Bring in humans, that's apples and oranges. Those differences have just increased dramatically.
Oh yeah, definitely. And I suppose I was sort of thinking on just a second ago on junk DNA. Did you want to address that real quickly? I mean, you don't have to go in detail. We haven't really talked much about that in this video, but we've got other videos on that subject matter. Just kind of just a brief response to people who think that most of our DNA, because that's something that is. Popular among a lot of evolutionists online is that they would try to argue that most of our DNA is not functional. I've heard Erica say that before, but in reality, I've actually know that scientists that are evolutionists will actually admit that most of our DNA is functional, but they will try to argue for degrees of functionality. Which, of course, you can imagine that different parts of our DNA is going to be more functional than others, but the point is that it does have a function to it. But a lot of critics online still propagate the idea that, I mean, as outdated as it is, they will still sit there and say that most of our DNA is non-functional. Would you say that's your assessment, that a lot of critics are still throwing that around, like Erica from Guts of Gibbon? I know that from what I understand, you've actually responded to Erica in a debate over that very issue where she thought that most of our DNA was junk, or that it was non-functional.
It's a great question. I mentioned earlier briefly in the presentation over why the activity associated with transcription and biochemical activity is not spurious, right? I talked about how if the majority of this activity, 80% plus, really was just noise or spurious, not functional or beneficial activity, then if evolution's true, evolutionary mechanisms like natural selection should have weeded out all of this genetic baggage, right? Because it would clutter up the system. It would clutter up the cell. It's wasteful of energy and resources for the cell. I mentioned how every time you add, Brian, a single nucleotide to a growing RNA chain, what are you doing? You're consuming energy. You're consuming ATP. And so if it really was just noise and spurious transcription, then there should be mechanisms today to suppress that noise, but there should be mechanisms in the past, like natural selection, that removes that baggage. Another thing has to do with transcription factors. Okay. Transcription factors, Brian, are proteins that regulate the transcription of genes. Okay. That is, they're copying into RNA on the way to make a protein. And transcription factors actually help to ensure that the right genes are expressed in the right cells of the body at the right time. Okay. And so here's the problem. If all of this really is junk and you have transcription factors just randomly binding all over the genome, what's that going to do? It's going to muck up the systems of the cell, and it would create what's called an interference problem that ends up gumming up operations in the genome. Another thing, and this is a point I've never seen them respond to, as far as I can remember, is there are only so many copies of a transcription factor that are going to be produced. And so if these transcription factors are merely binding randomly to many sites in the genome, then there's not going to be enough transcription factors available to actually do the binding that is necessary for these transcription factors to properly function. And so that's just a couple reasons. You know, we spent an hour roughly going over all the reasons. But again, transcription is an energy intensive process. And if these operations in the cell were mostly noise, this would be a waste of energy and resources for the cell. That leads me to conclude that this transcription, about 60%, brother, and the activity, which is about 80%, okay, it is functional activity. It's beneficial activity. It's real transcription. It's not just noise. And one other point I'll respond to, because Dr. Dan Stern Cardinal, he brings this up. He did a recent video responding to Discovery Institute. He also had a debate with Dr. Casey Luskin, and he points to what's called transcriptionally silent pseudogenes and ERVs. Okay. And he says, well, if they're not transcribed, then they're definitely not functional. They're definitely junk. But I have several resources, okay, several papers right from the conventional literature that shows now that even transcriptionally silent pseudogenes and ERVs are functional. One specific paper on ERVs that came out within the last few years talk about the benefits of transcriptionally silent ERVs in promoting variation. So no, even those examples, we can show functions. We can show functions for just about everything, Brian.
Right. And when I was sitting there talking to that evolutionist, Dr. Ian Korf, that I had on here for a little bit during the when I was doing the Topics of Controversy series, and we just got on the junk DNA argument, and he actually admitted that most of our DNA is functional. He just tried to argue for degrees of functionality. He even put the word junk in quotation marks when he talked about it. So I mean, it blows the mind, the fact that critics still would throw that argument around, knowing that the if you talk about professional scientific community, even evolutionists that may not even want to debate young Earth creationist or get involved in the origins issue, and they will sit there and admit most of our DNA is not junk.
Yeah,
And they'll admit that. Go ahead.
your militant evolutionists, they really need to hold on to junk, because the second they, the militant ones, the ones that battle us especially, The second they admit that the majority of the genome is functional, they're now admitting to a testable prediction, confirmation of that prediction for design diversity. We would predict that the majority of the genome is functional, right, for created heterozygosity. And they would be admitting that genetic degeneration is a real legit problem. Because the more the functional the genome is, the more deleterious all these mutations become. But if they have genomes of junk, what does that mean, Brian? Well, they could just say, okay, yeah, the mutation rate's high, but the majority of mutations are landing in neutral spots, non-functional spots in the genome, and therefore those mutations are strictly neutral. They're not really doing any damage. Notice this, brother. I shared screen. We could do a whole show on this one day. It's truly fascinating. While traditionally considered junk DNA, Dr. Dan Stern Cardinal, I hope you're listening, with no function, recent research indicates that even transcriptionally silent pseudogenes can play a role in gene regulation by acting as a sink for microRNAs, effectively preventing them from targeting and regulating their corresponding functional genes, thus indirectly influencing gene expression levels. And you can look to the functions of transcriptionally silent ERVs. And so, yes, all of these pseudogenes, ERVs, ALU sequences, they exist within the 80% of the genome that is active. And so that tells me that even the functions that we don't yet know for certain pseudogenes and ERVs, that tells me that there are functions. We just have to do more research. And so, as far as I'm concerned, brother, the junk DNA paradigm has been overturned.
There's a lot of creationists that would even consider this outdated and be blown away by the fact that there are critics online that still say that. And then it's like, why are they saying that? We live in the day and age where we actually know so much about our DNA that we know that most of it is functional. It's like that the idea that most of it's not functional is an outdated concept. And so, but you'd be surprised at how many people still propagate that. And a lot of evolutionists online, despite what they might promote, don't like talking about what they call junk DNA. Erica says she doesn't even like talking about it.
Yeah. Well, it's a difficult subject for them to argue that the
Right.
majority of the genome is junk.
And then
And
and they expect to have to demonstrate that because there's no way they can demonstrate it, even if you did not know the functionality of a particular DNA strand or whatnot. I mean, that does not necessarily mean it doesn't have one. You just don't know it.
right.
Uh, but
Yeah, I mean
go ahead.
a lot of genes, a lot of DNA elements, they are silent or turned off, right? There's a lot of ERVs that are functional during various developmental windows, during embryological development. And when they do their job, they're turned off. And so if they were to look at that ERV when it's turned off and not doing anything, they're going to conclude prematurely that it's junk. No, it's already done its job. And some of those can even be repurposed to do other stuff in the life of the organism. So it would require unlimited genomic sensitivity to really conclude that any genetic sequence is non-functional. They don't
Right.
have that kind of unlimited sensitivity to make that assertion.
I wish we had time to talk about ERVs, but we did a whole program on ERVs. But ERVs pretty much
That
fall
was
in
a
the
good
same
program
category.
too.
ERVs fall in the same
That was a
category as
great program. I enjoyed
yes, it
that.
was the same category as junk DNA because it's functional, and so
Yeah. Oh yeah. Anyways.
and I do have a question, a real quick question on that. Do you see that the fact that it has a purpose in it, a function, would that contradict the idea that this is that these ERVs, these what's they call extol or endo or was it exogenous? What's the word?
Endogenous. Endogenous. Yeah.
Endogenous, yes, right.
Yeah.
Endogenous retroviruses, and they came from like exogenous retrovirus, outside of the um, uh, from the outside to I think you said that
Oh,
it had
yeah.
the sperm line or something like that. It had to for inheritance.
Yeah, oh, yeah, yeah. Yeah. If you're looking at an endogenous retrovirus, that would mean like endo would be occur from within. And then an exogenous retrovirus, what that means is it's occurring from outside. So
And would
a good example of an exogenous retrovirus is HIV, right? That
right,
is transmitted horizontally. Endogenous, it's occurring from within. And so for it to be an endogenous retrovirus, it has to be an exogenous retrovirus that invades or infiltrates in a germ cell. That exogenous retroviruses, they're horizontally transmitted. Endogenous retroviruses, they are vertically trans or to get an endogenous retrovirus, you would have to have vertical transmission of an exogenous retrovirus through the germ cell line.
right. Okay. My question was on the purpose of the like you would see
Oh,
that
yeah, right.
it having functionality. Would that contradict the idea of this just being caused by a exogenous retrovirus that affected the line, germ cell?
A- Absolutely. Because in a nutshell, the question is this: are ERVs the ancient remnants of past viral infections, or are they created units of DNA function? Therefore, we as creationists, we would predict function if they're created for the most part. And evolutionists would expect non-function because these are just viral fossils, vestiges of evolutionary history. And so the fact that we find ERVs functional in determining cell types, functional in the immune system, functional in cell stress responses, embryological development, they're functional in tumor suppression. I mean, I can go on and on and on all day. So that tells me that yes, rather than being the ancient remnants of past viral infections, they are better understood as what? Created units of DNA function.
Some of these are like embryological development in it.
Yeah, there's one paper that I went over in our program together. Sure.
All right.
Literally admits that if we didn't have these ERVs, we could not exist. Mammals could not exist because we require them for placenta and placental and environment or embryological development.
Um, all right. As a last question, what looking at this, this is one of your primary focuses in on your channel is on the genetics, is on the genetic side. So in your study of genetics, in your study of all these different genetic arguments, what would you say is, what have you found that you would probably make a conclusion from that, like this is the genetic data, and this is what the Bible says, and this is what we actually found to be true in genetics, and things that we predicted that we would find, and genetics is a pretty new field, so it's not a study that's been around for a millennium or something. It's something it's fairly new, and we've been studying it, and the more we study it, and you got Dr. Georgia Purdom that I've talked to, and introduced something that's also a new field of subfield of genetics called epigenetics. And this introduces a new layer of information inside our DNA. I think that's kind of cool. You can have the same sequence of DNA, same letters and everything, but it could have a different message, if you want to call it that, depending on what epigenetic marker you have. So if you have a certain epigenetic marker, it might actually have a different information, a different role, a different purpose, a function, whatever the case may be. And even though the letters are the same, are exactly the same, She describes it like the way we would talk in our language and stuff, where you put a comma in a certain spot, and it changes what that statement means. And so that's to me that's just interesting that we have that based on our modern understanding of genetics, that the more we study genetics, the more it seems to confirm with the Bible. So I guess my question to you is that is that what you found, that the more you engage in genetics studies and that sort of thing, that the more you see confirmation of scripture?
Absolutely. Excellent question. As I went over in the beginning of the presentation, the Bible's clear. God created two people, Adam and Eve, just 6,000 years ago, roughly, separate and independent from any other form of life. That includes chimpanzees, orangutans, that includes bacteria, fish, whales. And so there are some very specific genetic related statements in what the Bible says in Genesis, that the earth started with two people, Adam, the first man, Eve, the mother of all living. And so, since the Bible claims to be the history book of the universe, we should be able to test what we know about empirical scientific data to the Bible's claims of history. And what do we find? Well, we find more and more, just as you said, that as we advance in science, as the years go on, we find more and more data confirming biblical history. And just a few of those that we've touched on during this program: mitochondrial DNA can be traced back to a single woman just a few thousand years ago. Haplogroups, right? The three major haplogroups, L, M, and N. All other haplogroups, all DNA diversity and mitochondrial DNA goes right back to those three women, Shem, Ham, and Japheth, and their wives. The Y chromosome, every single male Y chromosome on the planet, Brian, nearly identical and can be traced back to a single man. We know this based on DNA diversity. We know this based on the mutation rate. And the last thing I'll say, which we spent a lot of time on, which is great, the Y chromosome dissimilarity between humans and chimpanzees. When you consider overall architecture, size differences, and gene content, the human chromosomes are less than 30% the same. One woman, one man, and the data suggests that they are very different from even what the evolutionary community says is our closest cousin, the chimpanzee. And Brian, my brother, that absolutely decimates the secular view of history. It confirms biblical history, brother.
Absolutely. Have you ever studied epigenetics, by the way?
Oh, yeah, absolutely. Yeah, we've done videos on it. I brought it up. I used to throw the epigenetics reality. I used to do a lot of debates on modern day debate years ago when I first started doing this. And I was debating teachers and I was debating all sorts of serious students of evolution. I debated Erica 3 times. And one of my main points in cross-exam or discussion was epigenetics. How do you guys explain epigenetics? I mean, epigenetics is what? Forethought, the pre-existing ability to adapt, this extra layer of sophisticated information above and beyond your genetic code, right? You have millions of genetic switches in your genome just ready to be turned off given the right environmental stimuli. And so this is evidence of forward thinking, which points us back to the forward thinker. What evolutionary mechanisms can you put forth that have forward thinking capabilities? Because natural selection, Brian, it's not thinking about something 50 years, 60 years, 70 years down the road. Okay. It's not thinking about something thousands of years down the road. It's thinking in the short time. How am I going to survive today? Okay. But epigenetics, the epigenome, that is thinking ahead. It's kind of like, it's akin to the design world. Human engineers design vehicles with what? A spare tire. Oh, that's redundant. That's junk. No, because if you enter an environment where you get a flat tire, thank God you got the spare tire. That's forward thinking. which points back to forward-thinking engineers. The epigenome points back to the forward thinker who is who? God, the God of the Bible.
Absolutely. Thank you for giving me your time tonight. It's been a late one. We kind of had a late start though. All right. Thank you
Okay.
again,
Great.
Donnie. I appreciate you coming on to Apologetics 101. And do you have any final things to say to the audience?
Brian, it's been my pleasure, brother. I love joining you here on the program. I've been here several times before, and I appreciate your willingness to be comprehensive and to touch on some of the most important topics. I mean, it's my mindset that, hey, evolutionists, what's your best evidence? What are your best objections? ERVs, chromosome 2 fusion, getting into the world of genetics and Neanderthal DNA, let's say, and let's engage those. Let's engage and refute their best arguments, because when you get rid of their best, what's left? And what's left is a lot of the incredible data points that we covered today in terms of mitochondrial DNA, Y chromosome, the empirical scientific data, brother, that has literally confirmed biblical history and the account of origins in Genesis. And so it's always a pleasure to be here with you.
Yes, it is.
You're doing great work. And so I do want to encourage you to keep up that work you're doing to expand the kingdom, brother, and to uphold the truth of biblical creation.
Thank you, Donnie, for your taking your time to discuss these with us. And it's been great. It's been fun. I had a blast. Thank you again. And thank you for what you do at your ministry as well. And I will be linking Donnie's channel as well as his website in the description box below. All right. I know you got to go, Donnie. So we'll talk later. God bless.
Okay. God bless you, Brian. We'll chat soon, brother.
All right. Thank you for watching Apologetics 101. I hope to bring you some cool interviews pretty soon. I've got an interview with Mike Ord coming up tomorrow. Hopefully I'll have that video out by the following week. And then that'll be my last video before I go on vacation, because I'm going on vacation toward the end of March. And so after March, we'll try to... I might be doing a video sometime soon, but it'll probably be sometime after March on the heat problem and refuting the heat problem. Also have an interview scheduled with Dr. Carl Werner. I think that's scheduled in May, though, so it's a few months out. But I'll be starting to get more interviews in following that. It is always a pleasure to have Donnie and Matt on the Apologetics 101. I know it's been a long one. Part of that's because I combined their interviews because of an issue involved with the scheduling. Because Donnie, me and Donnie went back on a few different dates, and he ended up getting the wrong date marked down on his calendar. But anyway, it's okay. Everything worked out at the end, and I thought what Matt did was really good. It was also a nice preliminary for what Donnie did, because presented, so they kind of rolled over into each other. So despite them being separate interviews, they were very, very interconnected, so that it made for a very good video. So I hope you guys enjoyed it too. I enjoyed both of my interviews with Matt and Donnie. This was the first time Matt ever appeared on this channel, so that was an honor, because usually he doesn't have a whole lot of time lately, but he was able to give us some of his time and for this video, and so did Donnie, and we appreciate both of them. And thank you again for watching Apologetics 101. This is Brian signing off.